Hi, good afternoon, everyone. My name is Hamza Farouk. I am an infectious disease and virology clinician and a facilitator for the infection program for INTERYem. Thank you for joining us today for the inaugural INTERYem Infection Conference. We have some incredible speakers for you today for our sessions, and I want to thank them in advance for taking out the time out of their busy schedules to be here with us. Now, just some housekeeping information. The conference is being held on Zoom and also, and also live on YouTube as well. Just make sure. Oh, sorry. I think there was some feedback, so apologies for that. Just some housekeeping information. Conference is being held on YouTube and on Zoom. You can type INTERYem into YouTube to find the channel. Closed captioning should be available on your screens to help with understanding at what is being said. Now, please keep your cameras switched off and have your microphone on mute. The keynote speeches will be 45 minutes long, with up to 15 minutes for Q&As afterwards. There will be a Q&A session after each presentation, so please type your questions into the Zoom Q&A box during the talk. Now, certificates, for those who have registered, there will be an online link for a feedback form. These will be needed completed if you wish to have a certificate. Now, please make notes as you go along, as these will be needed for the feedback form. Now, as with any conference, there are multiple sessions, some of which may be more interest than others, depending on your practice. So you can identify which presentations are useful to yourself to attend. For a certificate, you do need to attend 75% of the sessions over the 2 days. And now, just a brief introduction to INTERYem. The work of the International Network for Training, Education and Research for Yemen began in 2014. INTERYem has training, education, and research as its three founding motivations. We know that Yemen's healthcare providers are dedicated and knowledgeable, but often lack opportunities to develop in areas that are needed to improve patient outcomes. We do believe that the future of Yemen's healthcare population resides in the capacity of its healthcare providers and the need for capacity building in infectious diseases to improve patient outcomes, hence the need to prioritize training in infection. INTERYem hopes to identify gaps in expertise and training, and then to fill those gaps by exchanging ideas, sharing expertise, and fostering relationships, in addition to a partnership with other countries as well. A key aspect of INTERYem's infection training is dissemination of the knowledge and skills developed by participants to other infection colleagues within Yemen and those who are unable to attend courses. The INTERYem Infection Conference has several objectives. The foremost is a desire to provide an update for infection healthcare providers or an overview to those new to the field. Now, as we know, COVID-19 is a priority for many in Yemen and throughout the world, and rightly so with the ongoing COVID-19 pandemic. Now, although the conference is not focused particularly as much on COVID-19, it is hoped that the knowledge imparted by the conference will be transferable to other infection issues. Furthermore, the conference will provide an opportunity to exchange information by practitioners and academics in infection from several countries. And this interplay will allow seeds of collaboration to exist. Finally, the infection conference will allow for the initiation of consideration where gaps in infection training may be amongst Yemen's healthcare providers, and start us to consider how to fill those gaps. This conference is in a series of conferences. Previously held conferences are the Public Health Conference and the Family Medicine Conference as well. Now, hopefully we'll be having some more future conferences with the INTERYem team. Now, to start off, I think I am a bit ahead of time, but it's a good idea to continue as we are. To start, I wish to introduce the next speaker, Mr. Ajaz Chowdhury, who is a trustee of INTERYem. So just to start off, Mr. Chowdhury is a chartered civil engineer and has served as managing director of ACOMS's European business, the world's largest multidisciplinary consultancy firm. In 2012, he moved to Qatar to deliver a major infrastructure program, and therefore he gained first-hand experience and understanding of Middle Eastern culture. He has proven project commercial and financial management skills, which he brings to INTERYem. He retired in 2020 to allow time for voluntary and charity work. It gives me great pleasure to introduce Mr. Ajaz Chowdhury.
Hi, am I on? Shall I start? Hello, I am one of the trustees of INTERYem, and I would like to welcome you all to this conference addressing the important subject of infection. As Hamza just said, this is the third in a series of conferences by INTERYem dealing with important medical subjects, with the key objective of informing and helping to improve medical practice in Yemen. These conferences bring together global experts in their fields focusing on INTERYem's priority areas. The priority areas are a selection of specialities key to improving the overall health and well-being of the Yemeni people. The first two conferences also, I'll just remind you, as Hamza said, the first two conferences dealt with the important areas of family medicine and public health, and the feedback we got from those was overwhelmingly positive. And for anyone who might be interested, they are available to view on YouTube. As most of you will appreciate, Yemen is facing challenges on many fronts, including food insecurity, a non-functioning financial system, and the collapse of public services. It has been estimated that half of Yemen's public healthcare facilities are currently not operational, while shortages in staff and medicines have left the other half only partially functional. It was out of this need that INTERYem was set up in 2014 by a small group of dedicated doctors as a non-partisan, non-sectarian NGO to help develop the capacity of the healthcare workers in Yemen. The ultimate aim of INTERYem is to improve the provision of healthcare for the people of Yemen. We are now in the process of registering INTERYem with the UK Charity Commission. The Commission regulates charities in England and Wales, and we are therefore required to work within the relevant UK legislation. As such, INTERYem's governance and due diligence is overseen by a board of trustees who are responsible for making sure INTERYem does what it was set up to do. We are also accountable to the Charity Commission and have to report on the work of INTERYem on an annual basis. Just a few words about INTERYem. I think Hamza mentioned this as well. INTERYem stands for International Network for Training, Education and Research for Yemen. And our mission is to facilitate collaboration between healthcare professionals from Yemen and the rest of the world. Specifically, our objectives, INTERYem's objectives are to promote and facilitate the education and training of healthcare professionals in Yemen, raise public awareness of health issues in Yemen, and support and enable health-related research on Yemen. Our aim is to facilitate long-term, sustainable partnerships rather than to focus on short-term gains. This will require trust and commitment from all partners. We know this will take time, but we are in this for the long term. We do not wish to work in isolation from the health context of Yemen, so our intention is to work with and within the frameworks of the current Yemen healthcare system and not set up structures outside the system. Ultimately, we aspire for a Yemen where communities prosper through good health and healthcare professionals excel and inspire Yemen and the world. So why is this conference important? Communicable diseases remain one of the main causes of mortality in Yemen. In addition to the worst outbreak of cholera in history, other diseases, including diphtheria and measles, have increased in the fragmented state. The weakness of public health surveillance has led to difficulties in identification and prioritization of many public health issues facing Yemen today, including communicable diseases such as cholera and diphtheria. Lack of vector control programs have also led to an increase in diseases such as dengue and malaria. Furthermore, endemic disease eradication programs such as those for leishmaniasis and schistosomiasis are not being implemented effectively as previously. Respiratory tract infections and diarrheal diseases remain amongst the biggest cause of mortality in Yemen. And the health situation, as you know, has now been further compounded by COVID-19. Antimicrobial resistance has gained further traction due to easy availability of antibiotics, exacerbated by the lack of doctors who may be vigilant regarding their prescribing. The effects of the shattered economy and conflict have also been felt on immunization programs. Training and continued clinical education of infection is therefore vital for Yemen's present and future aspiration of reducing harm from communicable diseases. So just to end, I'd like to thank Dr. Mubashir Chowdhury and his team, including the International Coordinator for INTERYem, Dr. Rahib Al-Azani, Dr. Nicholas Wong, Dr. Khalid Al-Shire, and of course the infection facilitator for INTERYem, Dr. Hamza Farouk, for their work in organising this excellent conference. Thanks also to all the speakers for giving up their valuable time and sharing their knowledge and experience with us. And last but not least, thank you to all the participants for supporting the conference and making our efforts worthwhile. On behalf of the trustees, I wish you all an enjoyable and successful conference. And on that note, I think I'll hand back to Hamza.
Thank you very much, Mr. Chowdhury. Now we will start off our first session of overviews infection. It gives me great pleasure to introduce our next speaker, Professor Janet Hemingway. Professor Janet Hemingway is a professor of tropical medicine at the Liverpool School of Tropical Medicine. She is also a senior technical advisor on neglected tropical diseases for the Bill and Melinda Gates Foundation and has 38 years experience working on the biochemistry and molecular biology of specific enzyme systems associated with xenobiotic resistance. Professor Hemingway has been PI on projects in excess of £200 million, including the BMGF funded Innovative Vector Control Consortium, the UKRI funded ICON Consortium, the ERDF funded Formulations Program, and the BMGF funded Visceral Leishmaniasis Elimination Program. Professor Hemingway was appointed the director of the LSTM in 2001 and stepped down on the 1st of January 2019, having overseen a period of exceptional growth of the organization. This included the awarding of the Higher Educational Institution status and degree awarding powers to the LSTM. Due to the incredible services, Professor Hemingway was awarded the Commander of the British Empire for services to control of tropical diseases. It's my great pleasure to introduce Professor Janet Hemingway. Oh, apologies, Professor, you may be on mute.
Yeah, thank you very much, Hamza. And I'm absolutely delighted to join the conference. It's around 12 years since I was last in Yemen. Even then, I think there were lots of issues with the health systems and health services in Yemen, and LSTM was delighted to link up in a long-term partnership, which, despite the current issues that Yemen is facing, has continued. And so it really is a pleasure to be part of this program and to give you a little bit of insight into some of the things that we're doing to actually try and speed up getting new anti-infectives to the market for countries like Yemen and for others globally. Many of them are required for the UK these days, given COVID is not an issue just for the low and middle income countries, but a global issue. So I'm going to try and share my screen so that you can see my slides. Okay, is that sharing? Can you see that?
Yes, we can, yes.
Great. I'll move it onto the slideshow. So what I wanted to talk about was infection in low resource settings, challenges and opportunities, and the way through the recently funded ICON Infection Innovation Consortium, we're trying to tackle some of these issues and problems.
Apologies, Professor Hemingway. I think we're seeing the presenter slides that are coming up
Okay.
rather than the actual.
So I have on my screen, it's just showing a single set. So I'm not sure how I now move out of that because it seems to be just going full screen. So I may have to take it all the way through and start it again.
Oh, the team are happy for you to continue in this way if you're happy with it.
Okay, it may be the easiest way of doing it because on my screen it's showing as a full presentation.
That's absolutely fine.
My apologies for that. I don't know quite why it's doing that. So the ICON Consortium was actually funded in September of 2020, but we obviously bid for that before the COVID pandemic hit, although we've been able to pivot some of what we're doing to handle COVID. And I think that really speaks to the comment that our earlier presenters were making about the skill sets that you have for infectious diseases generally being very appropriate when something new comes along, and where you can then utilize those skills very quickly to the benefit of the country. So just to give an overview of the talk, I'm going to talk a little bit about the anti-infectives landscape, introduce what we're doing with ICON and the objectives that we've got, what are our outputs, and how we are working with industry to actually make sure that these actually turn into real products that people can utilize within their own health systems. So this, for those of you who are already working in the anti-infectives landscape, will be very familiar. We know that it's a broken market, effectively. It's underinvested and needs new solutions. And a lot of the innovation is coming primarily from academia and small and medium-sized companies. That was very obvious with the vaccine initiatives that we've had for COVID, for example, where all of those initiatives came out of academia and where they were very rapidly partnered up with large-scale industry to take these through to a product that we can now all start and use. If we look generally at the anti-infectives landscape, whether that's antibiotics or drugs or vaccines, many of those products are at the end of their life cycle, and many of them are suffering from resistance starting to develop to them. So it's a basic, simple biological fact that if you use an antibiotic or a drug very effectively to kill large proportions of those organisms within the human population, inevitably at some point resistance will start and come through. And so we really need to make sure as we live with these diseases long term, that the pipeline of new products is robust. And so as one product fails, we have another product waiting so that we can continue to treat many of these diseases. The risk, as we've seen with antibiotics, is if that pipeline stops, we get to the point where the first line treatment is also the last treatment that we have. And as and when that fails, we have serious issues potentially coming through. But we all know that there is a poor incentive for the bigger companies to get involved in this market because the way we develop these products at the moment, they have a very high attrition rate. And the profit margin is very low for industry. And so there's very little incentive for them to get involved. Even pre-COVID, I believe there was a very different way of looking at this and a very different way of setting up this pipeline so that we could reduce the cost of getting new products to market, speed up getting those products to market, and make sure that they were accessible. And again, I think that's been demonstrated with the COVID vaccine, where a process that would normally have taken between 10 and 20 years has been truncated into 18 months to 2 years. So clearly, what I've been saying for a very long time is perfectly possible. And we shouldn't just be doing that for the COVID vaccine. We should be thinking about how we do that safely and practically for new drugs, new antibiotics, and new diagnostics as we go forward. And that really just says that. So the consortium that I've pulled together pulls together the different strands that you need to work together if we're going to do this. It needs the academics who are working in this area. It needs industry, both large and small, and it needs the health system and health service. With the ICON consortium, because of the way it was funded, it's our local health system within the UK, but the products we're developing are there for the global good and will be relevant to Yemen as well as many other low and middle income countries. It's set up at the moment as a £173.4 million programme with a reasonable amount of the funding, 18.6 million, coming from the UK Research and Innovation Council within the UK, 6.6 million from the European Union, and another 148 million from various other partners, including the Bill and Melinda Gates Foundation, other philanthropic donors, and large-scale industry. So we have partners like Pfizer, Unilever, GSK, and others involved within this programme. The idea is that we grow this program even further. So as I say, this has only been established since September, and we aim to have this turning over about £1 billion a year within the next 10 years to actually really sustainably get the pipeline for new therapeutics moving for everybody's benefit. How are we going to do that? Well, we have various objectives. We're going to enable through partnership, and that's working with small businesses, research institutions, and large-scale industry. And we will work with organizations wherever they are in the world, where they can bring ideas, information, and expertise to the partnership to try and get this global system moving. The idea is that we co-develop and de-risk new infectious disease formulations, but we don't stop at that point. We want to actually validate and position those operationally in the marketplace so that everybody gets the greatest benefit from these, and we make sure that we steward these products very well as they get into operational use. This is all about facilitating through open access. So we want these products available to everybody, even though we will have to have an end manufacturer for many of the products. But we are trying to get the products through cheaper, quicker, and smarter to the market. And in doing so and de-risking this for industry, are looking to industry to bring these into the market at a much lower profit margin than they might do if they develop these on their own. And that is a model that the Bill and Melinda Gates Foundation have been using successfully for quite some time. So this really is progressing products into the market from early stage discovery through into the clinic and even beyond that. In terms of outputs, we're actually also developing a number of new companies within this portfolio where there is clearly a market need, but something that the large scale industrial partners are not going to take through because the market is too niche. We're radically innovating product development, specifically around AMR, SARS-CoV-2 and insect-borne infectious diseases, but we're also dealing with some of the other respiratory diseases as well, as you'll see. It's important to the UK as well as globally. So the UK is backing this because it will have an economic impact here as well as the impact on health globally. And I've already mentioned the increase in funding that we look to be catalysing in this area over the next few years. So the basis of this programme is a series of open platforms that any company will be able to come and work with us on. Some of these platforms are physically located in the UK. Others are virtual platforms where we're putting expertise in but there isn't a physical platform, for example, around improving randomized control trials and getting those through the system far more quickly and cheaply than we do at the moment. We're tackling multiple issues, and I don't have time to go through absolutely everything in the talk today, but I wanted to highlight one or two things. So the first one I wanted to highlight was around antimicrobial resistance, an issue that is a problem for Yemen as well as many other countries, including the UK. And what we've got there is actually a very large natural product library. This has been developed in Liverpool with swabs taken from all sorts of different surfaces. It's been done as a citizen science project over about 10 years. So we don't mind what people swab as long as they tell us what they've swabbed, when they did it and where they did it. They send those swabs to us. We culture anything that is going to grow from there, and we culture them on bacterial plates with antimicrobial resistant bacteria to see if they are producing anything of real interest to us. And in the top of the screen there, you can see some interesting compounds, or at least one interesting compound there with M. luteus, where the sample that we've cultured is clearly killing the drug-resistant M. luteus there. We're then taking this huge library and fractionating it and putting it onto an industrial screening platform, which has been donated by AstraZeneca, and they're running that for us. It's a modular library so that people can come and screen samples that have come from soil or skin or home isolates, animals, although we're working with the Nagoya Protocol there, so clearly no animals that have been swabbed sort of without full agreement from the countries where the samples are coming from. We will then identify the compounds of interest as new potential antibiotics and identify those via mass spec. Those will then link through to companies working with us who can do the later stage development, and we will work with them on the clinical trials and hopefully get these all the way through to market. So that's just one of the platforms for antimicrobial resistance, which is the first one here under the develop column. But moving on from that, we're also establishing a hits to lead program to work with small companies and from anywhere in the world, where they can take what is their interesting early stage hit, and we can help take them through to a phase 1 clinical trial. As part of doing that, rather than following the old-fashioned methods of doing that, we're going to provide new humanized organoids that they can screen their molecules on, which will allow us to see whether they've got the right toxicology profiles and whether they are likely to be good candidates for taking into man before we put these anywhere near an animal or a human for further testing. And that allows us to allow things to fail early, fail quickly, and fail more cheaply than it would normally do. We're providing new rabbit and mouse models for screening where the organoid models won't work, and that's specifically against AMR pathogens that are very difficult to culture and very difficult to operate with. And those models will allow us to do at least animal pharmacokinetic and pharmacodynamic evaluations. We then move on to provide new human challenge models that allow safe and cost-effective first into man step, which is much smaller than a standard phase 1 clinical trial, safer, and the way that the COVID vaccine development program is going right now in the UK and various other places. Once we've actually got new products out to the point where they are ready to be marketed, we're also looking at defining the way that antimicrobial resistant pathogens are distributed through populations in different economic settings. And at the moment, we're working with the Ministries of Health in Malawi, Uganda, and the UK to look at where we've got commonalities and where there are differences. and then model what is the best way to use these new products so that we can keep the products working for as long as possible, but we also get the greatest health impact out of these products. So just to give you a little bit more of a flavour of a couple of those models that are really revolutionary, I wanted to talk a little bit about the human organoid screening. We're actually doing this screening in a BSL-3 lab, so that's a laboratory where you can clearly confine the infectious agent and work with it very safely, and setting up 3D cell cultures to allow us to test new molecules. Now, if you look at the standard way that everybody is screened up till now, this gives you the standard microtiter plate format where you would titrate drug, for example, in increasing concentrations on microtiter plates, have infected and uninfected controls and see what works. But that really is just one monolayer of cells that you're working with, and it's not a good surrogate for a human organ. So our human organ model, and this is going to be published in the next few weeks in one of the big journals. So you're getting this information out hot off the press, even hot before the press, is a 3D model for a lung where we can mix together basal-like cells at high purity from a mixed population of lung progenitors and actually in a test tube form something that functions very like a lung, even to the point where the surface layer on that has beating cilia in the same way that lung tissue would have. This is a much better way of screening new molecules, and because we can do this in a test tube or even in a microtiter plate as we turn this into a high throughput screen system, we believe it will be a much better way of sorting out potential new molecules as they come through. If we move on to the human challenge models then, we already have a number of these up and running. So we have one running for pneumococcal disease at the moment, and we have new vaccines from Pfizer and others that are being tested on that human challenge model. We have a working model for pneumonia, and we are developing models for RSV, influenza, and we hope multi-drug resistant TB and for COVID within the next 12 to 18 months. These operate in what many of you would recognize as a standard testing facility. So this is our expanded testing facility within the building that I work. It's an 18-bed facility, and this one operates as an outpatient facility, although we're developing a similar 18-bed inpatient facility for other trials. And when the COVID pandemic hit, we rapidly pivoted this to working on COVID, and we're actually the North of England site for the Oxford AstraZeneca COVID vaccine trial, vaccinating 10,000, I missed a zero off that, 10,000 participants, and we're following those people through, looking at the long-term impacts, if any, of the vaccination, looking at different age ranges, and as the new variants of COVID come through, are also ready to test the new variant vaccines that are set up to deal with those if they break through the current vaccines. Again, all of those we hope with the agreements that are starting to come through internationally will be made available globally. And our agreement between Oxford and AstraZeneca right from the start was that they would make these available to the global community at no profit to AstraZeneca itself. We're interested clearly also in looking at diagnostics, and we have two different platforms there. One is using electromagnetic sensor technology and getting engineers involved with that process. Because if you look at the vector control activities that we've been looking at, like indoor residual spraying, or even using insecticide-treated bed nets, to date, there is no quality control mechanism for those once they get deployed in an operational setting. And so what we wanted was a simple handheld device that could actually measure the amount of insecticide on a wall or on a bed net in real time, so that you could check whether your program was really working well or not. And we've had engagement of local companies to generate a final product there. That's going at the moment out for testing in India. And we have a further 3 million of Gates funding for new product lines using this sort of technology. And the new products that are coming through are not just looking at insecticides, but we're starting to measure parasites in peripheral blood without needing to take a blood sample, using a similar diagnostic. So again, we feel that this will be very helpful in many different settings. COVID has clearly also required us to rapidly develop new diagnostics for the COVID market. And again, we were very well placed to be able to do that. So we already were a FIND centre, FIND being the International Product Development Partnership for Diagnostics, and we were already a WHO accredited validation centre for new diagnostics. We got further accreditation from the UK so that we could produce data for MHSA registrations. And we've been working with a whole range of different companies to help develop many of the PCR tests and lateral flow tests that are now being used globally. And that just says that. I should have said that earlier. Alongside that, we've been interested in formulations that will inactivate COVID on different surfaces. That in part was funded by the Ministry of Defence in the UK, but the product is available for anybody and everybody to use, where we were looking at formulations, sprayable formulations that would rapidly kill effectively 100% of the SARS-CoV-2 virus, or I should say inactivate it rather than kill it, given that it's a virus. So that I think I've really covered, but when I say we've done this, this is obviously a very large consortium of companies, LSTM leading this, the local NHS hospital trusts, and it's been a great example of how if you get all of these partners working together for a common goal, you can very rapidly produce things that respond to global, national or international emergencies that come through from infectious diseases. But this doesn't just need to be something that is dealing with a pandemic response. We need to turn this into something that becomes the norm for developing new infectious diseases products. And I think if we get this right, that should be where we all start to go. Finally, I just want to say a little bit about optimizing product placement, because it's all well and good producing a new product, but that product will do nothing if you don't use it in the right place, the right time, in the right format. And so we've set up a very large 3-country study in Malawi, Uganda, and the UK, which is looking at public health surveillance, environmental surveillance of both AMR and now SARS-CoV-2, looking at how these organisms move from patients or people who are infected into the environment from shedding hotspots. And in the UK, those might be hotspots like a care home or a tertiary hospital. In Uganda or Malawi, that may be also happening, but you may also have those coming out from the farms where you've got, for example, a lot of antibiotic use for farm animals. And so we're looking at putting the technology into the countries that we're working with so that we can put a continuous detection system in place. And we've done that in Malawi already, where there is a system set up to do very rapid whole genome sequencing of bacteria and COVID shed into the environment. That information is generated on a Monday in Blantyre in Malawi. and the presence of any new variants, for example, are reported straight through to the Malarian President's Office the same week. So a good example of how we can get technology in a relatively resource poor setting moving so that it can help inform decisions for public health in the countries that we're working with. We're using other methods for enhanced detection in the environment in Liverpool. And again, modelling from that has helped shape the agenda for Public Health England and for the Environmental Agency within the UK in terms of how they're dealing with SARS-CoV-2. And then we're working with various companies if we know where the sweet spots are for trying to reduce this transmission so that we can work out what products ought to be used when to actually reduce the spread of these diseases and benefit the whole population going forward. And I think that's a really exciting program that more and more countries are starting to get involved with, and where we hope that there will be some generalized lessons that can go out for everybody to be able to work with. So I hope that gives you a flavour of some of the things that we're doing now that are quite new. It is all within the remit for the Liverpool School of Tropical Medicine, in that we're all about producing appropriate new therapeutics for low and middle income settings to try and improve health for everybody that we're working with. And we hope that everybody will benefit from these programs and feel that they can join in with the activities and where it is appropriate, move some of that technology back to their own countries. So I will finish there just by saying that we are still looking for partnerships. We are still open for more R&D collaboration, all the way through from clinical trials, right the way through to the epidemiology work that I've just described across the anti-infectives pipeline. And we would be delighted to see how we can expand the links that we've got with Yemen, because they haven't stopped even now, for everybody's benefit there. And I will finish there and open for questions if anybody has any.
That's great. Thank you so much, Professor Hemingway, for that very remarkable and insightful presentation. So just looking at the Q&A box for questions, I think the first one that we have is one of the participants is asking, the governmental healthcare sector in Yemen, of course, unfortunately, is paralyzed, and they need to establish medical and healthcare research centers to analyze all aspects and needs of infection control issues in Yemen. Would universities and departments such as the Liverpool School of Tropical Medicine, would they be able to help assist in establishing a project similar in this way?
Yes, I think is the simple answer to that. So as I mentioned earlier, we've been working with Yemen for many, many years and have continued to do so, even with the issues and problems that are sitting there at the moment. Obviously, at the moment, travel is almost impossible, but that doesn't stop us working at a distance. It doesn't stop us thinking about, as things start to ease up, how we look at these things, who might fund something. So what I don't have is a big pot of money to be able to say, yes, we can do this. But we are very open to collaborating to see how we can work together, identify the right sources of funding and take these forward, whether that's a one-to-one collaboration or as part of a consortium of multiple different countries.
Now, just moving on from the points that you made about travel, with restricted travel, it's a bit more difficult to actually be able to, to build collaborations. But now with the advent of telemedicine and teleconferencing, do you think there are some opportunities there that might help in setting up collaborations?
I would hope so. Clearly, we have still been talking with people right across the world as this problem has gone on, and maybe we can learn that we don't always have to jump on an aeroplane to collaborate and work together. That we can get things set up and get things moving as we go forward. I still don't think it will take away the need for one-to-one interactions face to face at some point, because often you get to know somebody, you get to trust people, you get to work and enjoy working together, and that's the way that long-term collaborations establish and keep going. So I think it's going to be a mixed model going forward, but certainly we're looking at how we travel less in Liverpool, reduce our environmental footprint, but still have a similar impact and still clearly work very closely with our overseas partners as well. So I think it's up to all of us to work out how best we can do this. And there won't be a single model fits everything, but we're very open to looking at how we can do that.
Completely understandable, and especially with the climate change occurring now as well, to reduce the carbon footprint, it absolutely helps, especially with things such as climate change driving, the propagation of other kind of vector-borne diseases as well. So no, definitely. Mm-hmm. Now, looking at the Q&A box, we've got one of the other panelists who said that, thank you, Professor Hemingway. They just wish to find out who is funding ICON. And is it a political organization? Would they be able to cooperate with the Yemeni government in North and South, and small NGOs like INTERYem, or just countries such as the United States and Europe? I think particularly with these political situations, Yemen, I think that is at the forefront of people's opinion. Yeah,
sure. So no, we are completely apolitical, and LSTM has worked very hard to make sure that that has always been the case. We don't get involved in anything that is politically driven. In fact, even when the US was being very difficult around some of its policies, we backed away from taking US money so that it didn't restrict us from who we could work with and where we could work until that eased up a bit. So this is not a political organization. It clearly is one that is focused on getting new products out. And I see one of the questions there is also, who will these benefit? I hope it won't come as a surprise to anybody that we are getting these products through for the global market. So the target product profile that we're setting up for the new antibiotics, drugs, or vaccines is such that it should be affordable anywhere in the world, and we should be able to take it anywhere in the world. These are not drugs for rich countries or antibiotics for rich countries, and we would not be part of anything that actually restricted them to the UK or Europe or the US, for example. It would be completely outside our remit.
No, absolutely. Especially with the AstraZeneca vaccine as well. The main thing for that was the fact that you didn't need a super cold chain, and it's more in development for low middle income countries as well. And even from a price perspective as well, we with the vaccine was to ensure that the cost was low for LMICs in specific forms. So yeah. No, thank you so much for that. Now, looking at the next question that we have. One of the participants says, when will we have all the nice new antibiotics? Will they not just be used for the more kind of high middle income countries? And they have said, the LMICs like in Yemen still have the old antibiotics as well. So I think it's just regarding, I guess, from a cost perspective, antibiotics.
Yeah, I think there are two things. I think that there's cost, but there is also an element that we need to deal with, often with the governments that we're working with, and I mean the recipient governments there, to make sure that they can import material in, or they can set up facilities to produce in country. And so sometimes the barrier is not, can we supply these things, but is the country willing to actually take these molecules, basically. And so I learned from what I was doing with the public health insecticides that you have to work all the way through to not only getting the product, but actually getting the product into operational use. And there are some surprising barriers that you as a scientist, I didn't think would be there until I tried to do it the first time. And I'm wiser and older now, and I know those barriers are there. And so what we will be doing as we start and move these along our pipeline is try and make sure that we can actually get them out where they really, the need is greatest, and make sure that we've got the right links through to the procurement agencies, however that's done, and the government's ministries of health in the countries that want to work with us and take these products forward.
No, absolutely. I mean, similarly with, when the artesunate came into well-being and it was approved by the WHO as well, it went to the LMICs in a very kind of rapid format as well, because it was approved by the WHO as well. So no, completely understandable from that perspective. Now, one of the other questions from the participants is, how can people in Yemen get involved with basic science research, as the facilities are not as adept as in England?
Mm-hmm. So I don't think we should try and overnight reproduce what we've got in the UK and Yemen. It isn't going to work. But what we can do is look at what are the technologies that would benefit the country most. And that's what I tried to give you that example of Malawi. So until 3 months ago, Malawi had no means of doing whole genome sequencing. But we identified that as something that would be really beneficial, so that information could flow. So they weren't sending samples out of Malawi to get sequenced somewhere else, and then it was taking a month for the information to get back. We've put that facility in, we've trained people up to be able to use it. It's been run in Blantyre, linked up with the big teaching hospital there. And that information, as I said, is flowing straight through to the president's office within a week. So I think that's a great example of saying, what is the technology that we ought to try and link up with and ought to try and help develop? What is the beneficial impact going to be for the country? And how do we then work together to try and get that into place? And what we can do depends on how strong the links are. Malawi was relatively easy for us because, again, we have a long-term 30-year link there. And so we had all the systems in place, we had all the contacts in place to move this forward, and we were able to identify sources of potential funding to get the equipment into the country quite quickly and get the training sorted out. Sometimes it will be a bit more slower than that, but all of these things start from something small. And so I would encourage people to think, okay, how would you like to get involved? How can we do that? And starting small will grow into something that is bigger over time.
No, I echo that. I mean, that's a prime example of how research has resulted in public health actions, because, all the whole genome sequencing reports reaching the public health teams there, the president's office as well. That way you can monitor the variants of concerns and see what needs from a research perspective, from a vaccine perspective, with the kind of variants as well. So that's a brilliant point. So thank you so much for that. So now the next question we have is that diagnosing infectious disease is very important in countries such as Yemen, specifically in regards of having point of care tests. These must be, as they've mentioned, to be inexpensive and relatively easy to use. From the participants' perspective, they're just asking what work is being done to further make further use of this.
Mm-hmm. So with all our programs, and the diagnostics one is exactly the same, we have a target product profile that says what is the price point that will allow these to be used in the settings that we would like them to be used in, and we develop the diagnostic to actually hit that price point. If it's going to be way more expensive than that, we don't develop it in the first place. And so all of these, we hope, right at the start, are set up so that they ought to be affordable. I say ought to be because, again, any of you who've worked in the health system know that there are always competing pressures. In Yemen, it's the same in the UK. So while it looks affordable, even if you've got a diagnostic that's way less than $1, that's $1 spent on a diagnostic that you could spend on something else and there's always a decision to be made there. But we do try and make sure that these things right from the beginning are set up in a format that they are usable by the people who need them. And we've also tried to make sure with the technology that although it can sometimes be complex technology, as I showed you with that electromagnetic wave detector, that uses some complex technology in terms of getting it set up. It's using artificial intelligence and machine learning, but the actual device itself is coming in at around $10, $12. And to do a quality assurance program for your indoor residual spray program, you need about 10 of those, and we've built them very robustly. So that device should last for years, as long as it doesn't get lost or anything else. And it's simple for anybody to use. You don't need to understand the technology. You put the thing onto the wall and there's a traffic light readout in terms of a red, amber, green, amber, red light. The top red being massively oversprayed, the next amber oversprayed but within a reasonable limit, green within the right tolerance, amber below you're undersprayed but there's insecticide there, and red at the bottom, no insecticide. And so you can get anybody to use that as long as they just understand what the lights mean. And you've got a device there that I think could save literally thousands of lives in malaria control, and is less than the cost of half a dozen bed nets, basically.
That's great. Thank you for that. There's a lot of interest in asking you questions, Professor Heminger. So there's our kind of chat box going through the roof. Now, one question that they have, well, obviously, I mean, the one question that they do have now, another one is that one of the participants has mentioned that there is the need to develop the medical education system in Yemen, because unfortunately the curriculum is a bit of out of date. Would there be any opportunities to kind of have this in the future with kind of organizations such as the Liverpool School of Tropical Medicine?
Yes, again, is the simple answer to that one. So, in fact, the reason that I was in Yemen all those years ago was that we were actually working with the curriculum then, working with all the major teaching hospitals and helping develop that. So we trained a cohort of, I think it was about 50 Yemeni doctors who are all back in Yemen, and we're still working with many of those, and they're still in contact with us. And so that is something that we've done in the past and would be happy to try and do again in the future.
Excellent. Now, the next question I have is, they've mentioned that the situation in Yemen is, of course, very difficult to currently work with any research, except if safe conditions allow for epidemiological work. Currently, even electricity and water supplies is very difficult with kind of the issues over there. From a research perspective, would there be any avenues that they could approach if some of their kind of electrical and water supply is kind of affected?
Beyond me, I'm afraid. So these are really difficult problems and issues, but I think that is a tough one. That potentially is something that the NGOs that are working there would be better placed than we are to try and help with.
Yeah, of course. I think it's just a perspective of the only thing that you can do is use point of care tests, as you mentioned. That's the only thing we can do, because I think there has to be a whole infrastructure change that needs to kind of occur. Now, just a couple more questions I think that we have. One thing that people mentioned: what are the main barriers and challenges to infection research in LMIC such as Yemen? And what do you think would be translating this into practice? What would be, in your esteemed opinion, the main challenges that you think are occurring?
Oh, heavens. I think that is a really tough question. And I think you're almost better placed to answer that than I am. So I would certainly look at where you've got the greatest amount of transmission with what, and how tractable that is in terms of being able to bring solutions in. So I would almost develop a matrix of looking at what are the problems I've got, major problems in terms of disease, what is in place at the moment, are there some relatively simple solutions that might be brought in. And I think the other thing I would always encourage people to do is not think in complete silos. Many of the things that you do will benefit multiple disease prevention programs, not just one. And so trying to look at things that are going to give you that broad benefit, I think would be useful. We're doing that, for example, in India right now with the visceral leishmaniasis program that I'm involved with. And a lot of what we're doing isn't just benefiting visceral leishmaniasis, but it's also benefiting their malaria, their dengue, and their filariasis programs as well. And so by getting that benefit across multiple diseases, the Ministry of Health is more likely to support it, sees the greater benefit from it, and will continue it after hopefully visceral leishmaniasis is no longer an issue, but some of the other diseases are. But we put the infrastructure in place that will allow them to pivot to those other diseases very quickly and very easily.
That's great. Thank you so much. I do have the panelists asking, would yourselves be happy to take a verbal question at all from one of the participants?
I see no reason why not.
That's great. I think I've spoken enough to be honest. So yes, of course. Go. I think we might have issues. Did we have anyone who wishes to ask a verbal question? I think I've got, yes, so go ahead. I think we might be having issues.
This is why typing in the chat is a bit easier.
Yeah, that's yeah, of course. So now I think the only other question that I think I've got so far at this moment is, in your expert opinion, how do you feel that the COVID-19 pandemic has affected antimicrobial research at the moment?
So I think there are possibly two answers to that. In one way, you might think it's set it back a bit, but in fact, I think it may longer term be beneficial. And the reason I think that is that a lot of the way that these organisms move around environment and things like that are very similar. So as I gave in the earlier example, we were doing things with AMR. We just added COVID into the tracking, but we didn't stop the AMR. And the fact that we can do both together and just shift the technology a little bit, I think is beneficial. So I think we should be able to look at both of these things going forward. And a lot of what we've learned about how to speed up the development of antibiotics or vaccines for COVID, we ought to be able to implement for AMR as well. And so again, I think the AMR community will benefit from that if we're clever about making sure that we don't just learn the lesson for COVID and then governments say, okay, well, now we don't need to do that. We'll just go back to what we were doing. Short term, I think the amount of funding that's available for AMR has reduced, but I think it will come back. So the problem hasn't gone away, and we don't yet have the solution sorted out. So it's in everybody's interest to make sure that that gets regenerated as soon as we possibly can.
Oh, yes, thank you for that. I mean, of course, the COVID-19 pandemic has affected funding for research worldwide as well. So that kind of makes AMR research a bit more difficult. So I think it would be to kind of see how things after the pandemic kind of quietens down, would be to see where we can kind of reallocate resources after the pandemic.
Absolutely.
Now, just a couple more questions have come up. One is, one of the participants says that the infectious disease is not only a problem for the infectious disease doctors in Yemen, it's a problem for all specialities and also the public as well. They do need to work together, but unfortunately they do not understand the problem as well as the infection doctors as well. So the participant is asking, how would we be able to find the opportunities to rectify this?
Mm-hmm. Not a simple one, but what we have done, because everybody has the same issue, and particularly for the general public, the reason we set up that swab and send program, as I explained earlier, was actually as a a community science program, and that allowed us to get everybody from junior school kids through to adults in industry, a whole raft of disadvantaged populations, because it's the kind of thing that everybody can do, and they have fun doing it, and they see the results coming out from it. But in doing that, we're also trying to get out the messages of why this is important. They've seen from their own backyard, they've taken a sample from their own backyard, and they've been able to pick up AMR organisms. So I think it's using different approaches, because if all we do is just shout at people and say, you've got to listen, eventually it just turns into background noise. And so I think there needs to be a different way of getting people engaged, a different way of getting them understanding, and getting them talking about this. And if you can get the community to believe that this is a problem, it may also filter into the health system as well. So you've got a top-down and a bottom-up approach to doing that. And that's certainly the way we've tried to look at it.
No, definitely. I think especially with programs such as the WASH program that was kind of instituted as well, it did help try and reduce infectious disease as well, especially such as cholera. Like even having a bit of patient education, public education helps reduce the burden of infectious disease quite rapidly as well. So no, I think the importance of science communication can't be overstated in these circumstances. Now, I think we've got a couple more questions. We won't take too much of your time, but I think this one is a very good one. They said, "Dear Professor Hemingway, thank you for visiting Yemen. Please, you are welcome to come back when it is safe. My question is, what is the point of having all these antibiotics, as people like the doctors don't use them properly, and resistance will likely rise again, especially with the new cephalosporins as well. Maybe the new antibiotics they need to be like the heroin, cocaine, and controlled by the governments." I think the question basically is regarding whether there needs to be an oversight by the government in these areas to see whether they can try and control antibiotics, which are the newer ones.
Yeah, we are looking at all sorts of options around this right now. That's one option, but it's very difficult to control like that. The other way may be to look at the way that we formulate these things, and see whether there's a way that we can try and somehow build in a mechanism that reduces the likelihood of resistance. And part of what we have already been doing is mapping out where we've got negative and positive cross resistance, because we may find, for example, if you've got a high level of resistance to one antibiotic, then those bugs are much more susceptible than the normal bugs to some of the new antibiotics. You use the new antibiotic, resistance starts to come to that. But if it causes negative cross resistance to your old one, you may be able to put in a regime of saying, if you use this one after this one and then this one, you can actually control the rate at which resistance is selected and balance it within the population. We haven't really been able to test that so far, because the pipeline of antibiotics has been so small that we just haven't had enough molecules to be able to do that. But we do need to try and think about how we can do this. Otherwise, we're in the global arms race forever of making sure that we develop new antibiotics on a permanent basis, because resistance will inevitably come through.
Thank you so much. I think that's the importance of antimicrobial stewards as well. Like even here in the United Kingdom, it's very difficult to police the newer antibiotics, no matter how much you try. So I think having antimicrobial stewardship programs, even in LMIC such as Yemen, would be a very good idea. Now, I hope you're not too tired, Professor Hemingway, but we're getting a lot of questions for you. So I'll just ask a couple more before we let you go on your way, if that's okay. I think I've had a couple, but to be honest, I can just summarize these kind of like 3 or 4 into one. Is that they're asking whether there is a plan to have a LSTM research collaboration partnership or a local division, such as how there is the Malawi partnership in Yemen in the future. Especially, I think there's some kind of specific universities that they've mentioned there as well. So I think the main, question there is whether there is any kind of plans in the future of the LSTM to have a local presence in Yemen.
Okay, so we're very careful with these in that we don't want to appear or function at all in any kind of colonial type setup. And so what we do is we always set things up as collaborative partnerships, and they're usually set up relatively small. So if I can just give you the example of really how the Malawi organization really developed. So 35 years ago, that was a research grant on severe and complicated malaria for 5 years from the Wellcome Trust for one of the senior researchers in LSTM working with the Ministry of Health in Malawi. At that point, Malawi actually didn't have any doctoral clinical training in Malawi itself. Everybody had to go out of Malawi to get their doctoral clinical training. Because Malcolm Molyneux had been there working on the malaria program for 3 or 4 years, and then that got renewed, when they thought about setting up a College of Medicine in Malawi, Malcolm was asked to work with the government in Malawi to help set up the College of Medicine. That became the collaborative partner. Wellcome then increased the amount of funding that it was given, so it now is the Malawi, Blantyre, Liverpool program, equal partnership between the College of Medicine and LSTM, and Wellcome has continued to fund it increasingly over many years. So we have around, I think there's about 320 staff there now, of which 90% are Malawians, many of who've come through the training within the College of Medicine. And we are linked up with Queen Elizabeth Hospital in Blantyre and providing the clinical service and clinical training there as well. And that's a great model that works for everybody. But it's not a model that you would go in day one and get Wellcome to say put 20 million into. They've worked out that this partnership really is working, it works for everybody, and they're providing most of the funding, not all of the funding. Some of it is coming from other organizations as well now. So I think all of these things start by a one-to-one collaboration almost, by somebody in Yemen for this in this example, and in LSTM, where there are mutual interests. If that grows and both of those partners have the ability to maneuver around the politics and everything else that gets involved with all of these things, then you can start and grow that to something much bigger. So there's no guarantee that these will work. And we've kickstarted some other things and they've worked for a while and then they decline. But others, as you hear, have been going for a very long time. And personally, I've had links in Malawi, Sri Lanka, India, and Cameroon, all of which have lasted for more than 30 years. And so those are very long-term collaborations with those countries. I've got other collaborations that have lasted for much shorter periods of time.
No, that's great. I mean, that's the thing. I think what needs to kind of occur is have a local kind of research group that you can have a collaboration and build that link in. And from that, you can have a more kind of mutual beneficial collaboration as well to kind of help in the future and continue with research. Yeah. Because I think it has to be local people who have to drive this, because they have the local know-how of how things work in there. Because, you know, coming from a country like England in the United Kingdom, we're not going to be aware of things that are occurring in Yemen. And whether there are any, you know, public, private, or governmental partnerships until we have local people there who are kind of give us the knowledge that's occurring over there. But yeah, I'll do absolutely. Is I think what I'll do is I'll just ask you. I think because we've taken quite a bit of your time. Let me just see if I think there might be one more question, and then I think we'll just let you go because you've been a great sport in answering all these questions. as well. So I think let's have a look. I think the only other thing that someone has asked would be basically that regarding kind of like the private sector and regarding making alliances with them in case that the government activities aren't able to unfortunately help out. What would be kind of in your opinion regarding the opportunities that can occur with public-private initiatives?
Again, we tend to put together the consortium that will work. And so again, the example I've given you with ICON is large and small industry, academia, NHS trusts. We've also got the public health system and the environmental agency and various ministries involved with that. And so we're really agnostic in terms of what that should like. I start from what is the problem that we're trying to tackle, and then work out who are the people that we really need around the table to make that solution happen. And then we work quite hard to try and put that solution into place. So again, there's not one easy answer, because some things you would have to do through the government, other things you could do through private agencies, some you might need both in. And exactly as you just said earlier, you need to understand the situation on the ground as well, so that you don't go blundering into a system and cause more trouble than you provide solutions with.
Perfect. That's great. Thank you so much for that, Professor Hamwi. I have a feeling that you will be getting quite a bit of emails from the Yemen team and from the University of Aden as well, because I think they want to kind of see if the LSTM would want to have a partnership with them. So I think you will become probably very busy even as you are at this moment as well. But what I will do is thank you so much for your time. I think we've taken quite a bit of your time. We've had quite a bit of questions as well. So thank you so much for doing this. All I want to do is thank you from the INTERYem team and the Yemeni participants, and hopefully they can have a partnership with you in the future as well. So thank you so much, Professor Hemingway.
Bye for now, and enjoy the rest of the conference.
Great. Thank you so much. Right. So moving onwards, I think we are a bit ahead of time at this moment. I think Professor maybe is delayed a bit and will be joining soon. So what we could do in this scenario is have a 15-minute break, and then we can basically reconvene at 4 o'clock as well. So I will, if you all stay on the line in about, we'll share the screen again for the program for the rest of the conference, and we'll meet again in 15 minutes time. Thank you so much. Hi, thank you everyone for coming back, and I hope you've all caffeinated with the incredible Yemeni Matari coffee. So now it gives me great pleasure to introduce our next speaker, Professor David Maybe. Professor David Maybe is a physician specializing in infectious and tropical diseases. After training in the United Kingdom, he went to work at the Medical Research Council in the Gambia in 1978 and was in charge of clinical services there from 1982 to 1986. He joined the school as a senior lecturer in the Department of Clinical Sciences in 1986, and he was made professor of communicable diseases in 1994. He was an honorary consultant physician at the Hospital for Tropical Diseases in London from 1987 to 2019, and was the head of the clinical research unit in the school from 1995 to 2002, and then from 2017 to 2020. He was also the director of the Wellcome Trust Bloomsbury Center for Clinical Tropical Medicine and Global Health Research from 1995 to 2018. His research interests are in neglected tropical diseases, in particular trachoma, for which he has been working on since 1984, and in yaws, in sexually transmitted infections and in the causes of fever. He's also a member of the Scientific and Technical Advisory Group of the WHO Department of Reproductive Health, and also the director of the WHO Collaborating Center for the Prevention and Control of Sexually Transmitted Infections at the school from 2014 to 2020. He currently chairs the Scientific and Technical Advisory Group of the WHO Department of Neglected Tropical Diseases, and is a member of the WHO Global Alliance for the Elimination of Trachoma, and has sat on the Trachoma Expert Committee of the International Trachoma Initiative. He was subsequently awarded the CBE in 2014 for services to health development in Africa and Asia, and the Prince Mahidol Award for Public Health in 2019 for his work on trachoma. So, Professor Maybe, thank you so much for joining us. I will leave the floor to you, sir.
Thank you very much, and thanks for the invitation to talk. It's a great pleasure. I've never been to Yemen, but of course I read about it a lot. So thank you for inviting me. Now, I guess I need to share my screen. See if I can do that.
Yes, please.
Here we are, I think. Yes, can you see that?
Yes, we can.
Okay, excellent. So, going to talk for the next half an hour or so about neglected tropical diseases, which are, of course, old diseases, but we have some new approaches, and we have a new roadmap for 2030, launched in January this year on World NTD Day. So I'm going to talk a bit about some new treatments that have been developed since the last roadmap was put forward in 2012, new diagnostic tests, and the fact that we now need integrated strategies as promoted in this new roadmap. So what is a neglected tropical disease? These are the WHO criteria. It must disproportionately affect populations living in poverty, cause important morbidity and mortality, including stigma and discrimination in these populations, primarily affects populations living in tropical and subtropical areas, is amenable to broad control, elimination, or even eradication through what WHO call preventive chemotherapy, which I would prefer to call mass drug administration, mass treatment, or through intensified or innovative case management, vector control, or a One Health approach involving veterinary public health. And finally, it must be relatively neglected by research. This is the current WHO list. There are 20 diseases on the list at the moment. And the top 6 here, the control or elimination strategy is mass treatment, mass drug administration, or preventive chemotherapy, as WHO call it. And then there are others down here where you have to identify individual cases and then treat them. So the mass treatment, where do these treatments come from? Just a little bit of history here. So in 1975, the World Bank started a campaign to get rid of river blindness, onchocerciasis, which I believe you do have in Yemen or used to have. And this program initially was based on vector control, spraying the breeding sites, the fast-flowing rivers in which the black fly vector breeds. And then the same year, as it happens, a new drug was discovered, ivermectin, by a Japanese scientist and an American scientist who shared the Nobel Prize a couple of years ago. And this was developed initially as a veterinary drug to treat parasitic diseases in cattle. But in 1982, it was shown that it eliminates the microfilariae of onchocerciasis from the skin of humans. And 5 years later, ex-President Jimmy Carter persuaded the chief executive of the drug company Merck to donate ivermectin or Mectizan, which is the trade name, to the onchocerciasis control program. And he agreed to donate it for as long as it was needed. And since then, hundreds of millions of people have been treated annually with ivermectin, and this has had a huge impact, so that now very few people are going blind from onchocerciasis. So in the 1980s and 1990s, other studies were published. Trial of ivermectin and diethyl carbamazine for lymphatic filariasis, and a trial of a single oral dose of azithromycin for the treatment of trachoma, which previously had been treated with 6 weeks of twice daily tetracycline eye ointment, which was clearly very difficult to complete. So then in 2012, there was a meeting in London, where I am, at the Royal College of Physicians, chaired by Bill Gates, who you see sitting in the middle here, and attended by Margaret Chan on the left, Director General of WHO, representatives from the World Bank, UNICEF, UNDP, several major non-governmental organizations, and government representatives from a number of countries. And the London Declaration was published after this meeting, in which these people, in particular the chief executives of the drug companies, promised to donate their drugs for as long as they were needed for the control of NTDs. So shortly after the the London Declaration was published, the World Health Organization published their first roadmap for NTDs. And they targeted 2 diseases for global eradication, yaws and guinea worm, a number of other diseases for elimination from defined geographical areas, such as onchocerciasis from South America, and some other diseases for elimination as a public health problem, whatever that means. So specific targets were set for trachoma, leprosy, visceral leishmaniasis, and lymphatic filariasis. You will notice here that a number of targets were set for 2020, for last year. So what progress was made since 2012? Well, huge progress was made. So mass drug administration coverage for population at risk of these NTDs increased from 42% to 65%. So that for the last 4 or 5 years, more than 1 billion people have received mass treatment for NTDs each year. As a result, 500 million fewer people now require interventions, and 43 countries have eliminated at least one NTD. In 2019, the Scientific and Technical Advisory Group of the WHO NTD department, of which I'm a member, was asked to consider the development of a new roadmap because 2020 was only 1 year away. So a new roadmap for 2030 was being planned. And we said that this new roadmap should contain measurable targets, which should be ambitious, evidence-based, and realistic. We should identify roles and responsibilities at subnational, national, regional, and global levels, and in particular, encourage country ownership of these programs, which often had been led by international organizations in the past. We pointed out that eliminating NTDs was an essential aspect, was essential to achieving universal health coverage, which was named in the newly published Sustainable Development Goals. And it is essential that we encourage multi-sectoral engagement and an overall monitoring and evaluation framework so we can measure progress. The draft roadmap was put out for extensive consultation online and with regional and national stakeholders, so that several hundred people were consulted over the next couple of years. And finally, these targets were agreed at the end of 2020. A World Health Assembly endorsed these recommendations that we hope to see by 2030 a 90% reduction in the number of people requiring interventions against NTDs, 100% of the population at risk should not have to pay out-of-pocket payments for treatment of their NTDs. 100% of endemic communities should have access at least to basic water and sanitation. 100 countries should have eliminated at least one NTD, and those same two NTDs, yaws and guinea worm, should have been eradicated globally. And the number of disability adjusted life years lost to NTDs should be reduced by 75%. So yaws and guinea worm targeted for global eradication by 2030. Human African trypanosomiasis due to Trypanosoma gambiense should be eliminated from at least 15 countries, leprosy from 120 countries, onchocerciasis from 12 countries. Trachoma should be eliminated as a public health problem from all countries. Lymphatic filariasis and visceral leishmaniasis should also have met those targets. So what do we need to eliminate or eradicate an infectious disease? Well, clearly we need an effective intervention. This could be a vaccine, it could be mass drug treatment, it could be case finding and treatment, or in the case of guinea worm, simply health education and improving water supplies. So that's the first requirement. Secondly, we need diagnostic tools so that we can identify those last few cases. They have to be sensitive enough to do that, and they have to be specific, highly specific, so we don't keep getting false positives which make us think we haven't met our target. So these tests must be highly sensitive and specific and must be widely available. And finally, there should be no animal reservoir. So yaws is like syphilis caused by Treponema pallidum. And I believe in Yemen, you never had yaws, but you used to have endemic syphilis, which is also a non-venereal treponematosis. So a disease spread by skin-to-skin contact caused by Treponema pallidum, causing skin and bone lesions, such as you see here. This was the first disease targeted for eradication by WHO soon after it was set up in the early 1950s. And the strategy at that time was mass treatment with injectable long-acting benzathine penicillin. So mass treatment, or treatment as the prevalence went down, treatment of cases and their contacts. More than 50 million people received this treatment with injectable penicillin. The prevalence was reduced by more than 95% globally, a remarkable achievement considering these cases were mostly in remote rural villages in Africa, Asia, and South America. But by the early 1960s, there were not many cases being reported. Surveillance was discontinued and the disease came back. So in 2012, this Dr. Oriol Mitja, a young doctor from Spain, who had recently been on our diploma course in tropical medicine here at the London School, he got a job in Papua New Guinea, working for a mining company, and he found there was a lot of yaws in that community. So he did a randomized control trial comparing the standard treatment with injectable penicillin with a single oral dose of azithromycin, and he showed they were equivalent. This paper was published in The Lancet in 2012. The following year, WHO convened a meeting in a village near Geneva called Morges, and they came up with a new plan for eradicating yaws by 2020, rather ambitious as the meeting was only held in 2013. The new strategy was mass treatment with a single dose of azithromycin given by mouth, then subsequent 3 to 6 monthly surveys at which cases and their household contacts were treated. And once no more cases were found, post-elimination surveillance, doing serosurveys among young children to make sure transmission is no longer occurring, using these simple point-of-care tests, serological tests for syphilis or yaws. So it seemed we had the tools, we had the single dose treatment and the simple, affordable point-of-care diagnostic test. But there were still a number of challenges. So not much data on where yaws was still a problem. Many countries that used to report cases were no longer reporting them. Was that because there were no cases or because they had stopped looking for them? At that time, there were no funds available. Clinical signs are not very specific. We and others discovered that many children whom we thought had yaws had other skin infections. And then resistance to azithromycin was reported in Treponema pallidum, initially in patients with syphilis and then in patients with yaws. And it also seems that there is almost certainly an animal reservoir of Treponema pallidum among non-human primates, monkeys in Africa. So this target of eradication of yaws by 2020 was not met. What about onchocerciasis? So the intervention here, as I mentioned earlier, mass drug administration with ivermectin, which kills the microfilaria in the skin, but not the adult worms, which continue to produce more microfilaria. So you have to continue with annual mass treatment for at least 10 years, which is the lifespan of the adult worm. And the traditional diagnostic method is a skin snip. So you take a small piece of epidermis, put it in saline, look down your microscope a few hours later for microfilaria. Clearly, it's difficult to do in the field, and you can't really test large numbers of people in this way. So, what are the criteria for elimination? Absence or near absence of infective larvae in the black fly population means you have to test 6,000 black fly in each endemic focus. You can pool them in pools of 200 black fly and do a PCR test. And in children, you need to test 2,000 children to make sure the prevalence is less than 1 per 1,000. And to do skin snips on 2,000 children would not be feasible, but we now have a new serological test, so we can collect a drop of blood, use a dry blood spot, and do serology to an antigen of Onchocerca volvulus. Great progress has been made. Onchocerciasis has been eliminated from almost every country in South America. There's a small focus still on the border between Venezuela and Brazil. It's been eliminated from some foci in Uganda, and global morbidity has been greatly reduced, so very few people now go blind from onchocerciasis. There is a problem. If you look at this map of Africa, the red areas have not yet started ivermectin mass treatment. Why is that? Because lower is also endemic in those regions. This is the lower adult worm crossing the eye. In people with high loads of lower microfilaria, if you give them ivermectin, can cause a fatal encephalitis. So it has not been rolled out in these areas. But we have a new way to diagnose lower simply using a mobile phone, drop of blood, and you can detect high microfilaria loads with a mobile phone with an attachment, and therefore decide who should not receive ivermectin. So again, this is a very promising new diagnostic test. Moving on now to trachoma, a particular interest of mine, which I believe used to be endemic in Yemen. I'm not sure if it still is. So it's caused by Chlamydia trachomatis spread from eye to eye. This is my colleague Robin Bailey, who worked with me in the Gambia in the 1980s and did the first trial showing that 1 dose of azithromycin by mouth is an effective treatment for trachoma. After this, the manufacturer Pfizer were persuaded to donate azithromycin for trachoma control programs. And they have up to now donated more than 900 million doses. The target for trachoma is elimination as a public health problem from every district in the world. And the strategy is annual mass treatment with azithromycin until the prevalence of follicular trachoma, which you see here. So to diagnose follicular trachoma, you evert the upper eyelid and you look for these little white spots, which are follicles. If you see more than 5 follicles, that person has follicular trachoma. So you continue mass treatment until less than 5% of children aged 1 to 9 years have TF. And you then continue surveillance for 3 years after that target is met. Second target is that less than 1 per 1,000 total population should have trichiasis. This is the late stage of trachoma where the eyelashes turn inwards and scratch against the cornea. So these targets are based on clinical examination. It can be a little subjective, deciding if somebody has 5 follicles or 3 follicles in their subtarsal conjunctiva. You need well-trained trachoma graders who have been shown to pass a test of inter-observer agreement. But of course, as the prevalence of TF goes down, it's difficult to train your graders and make sure they're able to diagnose it properly. And secondly, not all cases of follicular conjunctivitis are due to trachoma. So you may find cases due to something else, which make you think you need to continue with mass drug administration when you don't really need to. So is there a simple diagnostic test for exposure to ocular chlamydial infection that could help you decide whether you have eliminated or whether you have interrupted transmission? These were studies that my group did in the Pacific Islands. So the Solomon Islands on the left and the islands of Kiribati on the right here. So in both these countries, the prevalence of TF was about 20% in children aged 1 to 9. But interestingly, in the Solomon Islands, there was very little trachomatous trichiasis, whereas in Kiribati, there was more than 10 times as much. So we wondered whether the follicular trachoma we found in the Solomon Islands was really due to trachoma, to chlamydial infection. So this is seroprevalence by age. You can see that up to the age of 15, almost no children have antibodies to chlamydia trachomatis. After that age, of course, it goes up, and this is due to sexually transmitted chlamydial infection. In Kiribati, there was a very different picture with almost 80% of 7 to 9-year-olds being seropositive. So we think this simple serological test, just using 1 drop of blood collected onto a dry blood spot, may be helpful in deciding when we can discontinue mass drug administration for trachoma. Huge progress has been made. The Global Trachoma Mapping Project was completed in 2016. This was the largest disease mapping project ever undertaken. 2.6 million people were examined in 29 countries. So we now know exactly where trachoma is a problem and where to focus our efforts. Since 2002, we believe the population at risk of trachoma has been reduced from 1.5 billion to 137 million, and the people needing surgery to turn those eyelashes out again has been reduced from more than 8 to less than 2 million. 11 countries have been validated by WHO as having met those elimination targets. And interestingly, a study done by colleagues of mine in 3 African countries showed that mass drug administration of azithromycin reduced all-cause under 5 mortality in this study by 13%. So although mass drug administration may select for antimicrobial resistance, on the other hand, it has collateral benefits in terms of treating other infections and, as shown here in some settings, reducing all-cause under 5 mortality. So these are the latest predictions on the number of people needing interventions for the control of trachoma. So we believe that at the present rate of progress, we will have eliminated trachoma globally as a public health problem by 2030. Moving on now to consider human African trypanosomiasis. So treatment for this is, or until recently, has been difficult. Intravenous treatment for at least 10 days and toxic. It occurs in mainly remote rural communities in Africa. Diagnosis traditionally is by a blood film. You see the trypanosomes here. 20 years ago, a simple microagglutination test was developed for Gambiense trypanosomiasis, which you see being done here, but clearly you need electricity, you need a fridge to keep your reagents, and it's not easy to do this in the field. So control, elimination depends on finding the cases in those remote rural villages, and the treatment you give until 2 years ago depended on the stage. So was the central nervous system involved? If so, you had to give intravenous treatment for 10 days. And in the case of Trypanosoma rhodesiense infection, the treatment was with melarsoprol, which is highly toxic arsenic-containing drug. In recent years, we have made considerable progress. We now have a simple lateral flow assay, a point-of-care test for Gambiense trypanosomiasis, which is highly sensitive and specific. And 2 years ago, the result of this trial done by Médecins Sans Frontières in remote villages in Central Africa showed that this new drug, fexinidazole, given by mouth, appeared to be effective even if your central nervous system was involved. So this would mean you would no longer have to do a lumbar puncture in the field, and you could give oral treatment. So this is a huge advance. And great progress has been made so that last year, less than 1,000 cases of human African trypanosomiasis were identified and reported. Finally, in terms of new treatments, Buruli ulcer, endemic in parts of sub-Saharan Africa and Asia, caused by Mycobacterium ulcerans. Until recently, treatment needed 8 weeks of streptomycin injections daily plus rifampicin. This trial, done by Professor Phillips in Ghana, identified an oral regimen, rifampicin plus extended release clarithromycin. So this again is a huge step forward. The new roadmap calls for integrated approaches rather than vertical programs for each NTD. Can we combine our programs? Multisectoral collaboration, country ownership, rather than having outside NGOs coming in and delivering care. And we should emphasize that the Sustainable Development Goal of universal health coverage depends on controlling NTDs. And we need to measure the impact of our interventions. So in the past, these programs have tended to count the number of treatments given, but not necessarily measure the impact. We really need to measure the impact to assess progress towards meeting those targets. So for the integrated approaches, WHO have developed a framework for the integrated control of NTDs affecting the skin, such as yaws, leprosy, borreliosa, and scabies, which has recently been added to the list. We need new diagnostics that meet those criteria, being highly sensitive, specific, and available. So they have set up a diagnostics technical advisory group for the NTD program in WHO. Collaboration with interventions to provide water and sanitation and with global vector control programs is clearly important. And when we're giving out mass treatment, why not give treatment for more than one NTD at a time? We know that azithromycin is effective against yaws and trachoma. Ivermectin is effective against onchocerciasis and soil-transmitted helminths and scabies. But why not give ivermectin and azithromycin together? And there have been studies showing this is both safe and effective. And finally, if we could develop diagnostic tests that could be done on the same drop of blood on a dry blood spot for multiple NTDs, this would simplify measuring progress towards those elimination targets. So the new roadmap was launched officially in January this year on World NTD Day. And if you would like to read more about this, I recommend this special issue of the Transactions of the Royal Society of Tropical Medicine and Hygiene, which addresses that roadmap and has all you need to know about how those targets are going to be met. And that's all I have to say. I'm going to stop sharing my screen and will be very happy to take questions.
Thank you, Professor Maybe, for that brilliant and fascinating lecture. I think we do have quite a bit of questions. The one question that we do have is pretty much what are the main barriers and challenges to infection research and specifically to neglected tropical diseases in LMICs such as Yemen and translating this into practice?
So I think, I guess what we need to know is which diseases are still endemic in Yemen. I believe you used to have trachoma and onchocerciasis and possibly, I guess, soil-transmitted helminths, leprosy. I'm not sure which others, because as I say, unfortunately, I've not been to Yemen. But I guess, obviously you need to know which ones are still endemic, where they are, which populations are at risk, so that you can roll out these interventions. So I guess, I think the integrated management of skin NTDs, the new WHO manual is very useful. My colleague, Michael Marks, or several of my colleagues, Michael Marks and Steve Walker, have been doing this in West Africa. They've been training community health workers to try and diagnose these diseases so they can identify who needs treatment and which communities in particular would benefit from mass treatment, for example, for scabies, which I'm sure you also have in Yemen. So I think perhaps the focus on training community health workers to diagnose those skin diseases. Other thing that we've been using or exploring use of is telemedicine. If you can send a picture of the skin lesion, to a dermatologist, then that can be helpful in training your community health workers.
Excellent. Thank you so much. But from that, there's a point that leads on to our next question. With telemedicine and teleconferencing, do you think there's going to be more opportunities for international research in regards to neglected tropical disease? And specifically, you've mentioned the LOA scope as well. So if you could talk more about it, that would be very useful.
Yes. Well, I don't think you have lower in Yemen. Uh, so I think that's one thing, one entity you don't need to worry about. But I do think telemedicine is the way forward in many settings. I'm not a dermatologist, but we could discuss with my colleague Steve Walker as a dermatologist who works mainly on leprosy, and it might be worth discussing that with him.
Okay, excellent. Thank you for that. I think we do have a couple more questions from the Q&A panel. The one question they said, well, it's a comment and a question is, thank you, Professor. Maybe please do come visit us soon in Yemen when it is safe. My question is that there is a war currently in Yemen, and this has caused quite a bit of problems in the fight against NTDs. In this regards, would you have any opinion as to how we would be able to help fight this problem and this occurring?
Sorry, I didn't quite catch which disease that was.
Oh, neglected tropical diseases.
NTDs in general.
Yes. Yeah.
So which ones? I mean, I've mentioned some that I think you probably do have or used to have: trachoma, leprosy, onchocerciasis, scabies that's recently been added to the list. I guess snake bite. Do you have snake bite? It's another recent addition. I think, and then possibly soil transmitted helminths. I'm not sure.
Yeah, I think.
So I think we have the interventions. For most of those, ivermectin for the soil transmitted helminths, onchocerciasis, scabies, azithromycin for the trachoma. Leprosy obviously is a challenge to diagnose. And again, my colleague Steve Walker is the man to talk to you about that.
Okay, excellent. Thank you so much for that. The next question that we have is that they have said the mass eradication is absolutely required for yaws. The only thing it comes across is with similar to Professor Hemingway's lectures regarding antibiotic resistance. This is a big problem. So how would we be able to balance up mass eradication and stop antimicrobial resistance?
Yes, it's a challenging question. So we were I and my colleagues we led the way in promoting mass treatment with azithromycin for trachoma, and we came in for a lot of criticism from people who said you're selecting for antimicrobial resistance. And we have looked at this in trachoma endemic communities. We've taken nasopharyngeal swabs to look for pneumococcal carriage of strep pneumoniae. And we have found that for 2 or 3 months after treating the whole community, you will find some resistant isolates, but by 6 to 12 months, they've disappeared because there's not the ongoing antibiotic selection pressure just from one round of mass treatment. And as I said, azithromycin has collateral benefits. So certainly in those settings, especially in very poor rural communities in Niger in particular, in sub-Saharan Africa, it had a significant impact on child mortality. It has some antimalarial activity, it's broad spectrum antibiotic, so it's going to treat pneumonia and potentially salmonella infections and so on. So it is a balance. We shouldn't indiscriminately give mass drug administration unless we have a good reason to do so. I agree with that. And if possible, should try to monitor antimicrobial susceptibility.
No, that's great. And specifically, here point of care tests are very useful as well in these situations. Now, I think moving on, one of the participants has kind of prompted a question from this, which is praziquantel, azoles, and ivermectin are very good drugs. Have you, in your experience, have you noticed a lot of resistance with these medications at all?
That's a very good question. I'm not sure anyone is looking very hard for that. But, I mean, we've been discussing this. I mean, I'm not particularly involved in onco or schistosomiasis control programs, but it seems those who are say there's no evidence yet of resistance. Do you have schisto in Yemen? Schistosomiasis? I guess you may do. It used to be very common in Egypt. I know.
I think there is a bit of schisto in Yemen at the moment. I think some of the attendees have mentioned that. Now, moving on, thank you for that. I think the next one they have, question-wise, is one of the participants has mentioned that their opinion is that vaccines are better at stopping the disease from occurring, and they are better at treating afterwards as well. So I think they just wanted your kind of expert opinion on whether there's any work being done on vaccines for NTDs.
Yes, that's an interesting question. So I and my group have been working for about 30 years trying to develop a trachoma vaccine. But so far, unfortunately, without success, there is a candidate now being tried out. So I first became interested in trachoma when I was working in the Gambia, where they had done 3 trachoma vaccine trials in the 1970s, but they didn't work. So, now, I mean, now that we can, we've developed COVID vaccines in 12 months. So, perhaps with new technology, we'll have more success. But so far, I mean, in terms of vaccines, so, BCG vaccine has some protective efficacy against leprosy. That was shown in Malawi by my colleague Paul Fine some years ago. But I can't think of another NTD for which we have a vaccine. I guess if we had a vaccine, they wouldn't still be such a problem.
Yeah, true, definitely. No, I think similarly, I think with mRNA vaccine technology coming into, I guess, relatively widespread use due to the COVID-19 pandemic, I think we should be getting more candidates, and especially with CRISPR-Cas9 technology being built in as well, we should be able to kind of help in that aspect. And I guess similarly, the malaria vaccine is it's only come into being now, and they will be doing trials in the future for that. So we'll see how things kind of pan out for that.
Yes.
Now, the next question that I have is, I'll be honest, I'm not aware of this, but apparently one of the participants has said that the Morgus pan in Papua New Guinea is interesting. Was it successful, and what can it be of use in Yemen? Now, may I ask, are you aware of the Morgus pan by any chance?
Yes. I did mention that in the talk. So that was my colleague Oriel Mitchell, who in Papua New Guinea showed that a single dose of azithromycin is effective against yaws. And then WHO had this meeting in Morges, and that's what's now recommended. And it's, again, targeted for eradication on the 2030 roadmap. But I think there are big problems, to be honest, because we know that Treponema pallidum can become resistant to azithromycin, and that has been shown by that same doctor who did the original trial in yaws cases in Papua New Guinea. So I'm not sure it's going to be the answer for yaws or endemic syphilis.
Excellent, thank you for that. I think the next question we have is that the mobile phone diagnosis, so I think they're mentioning over here the lowest scope, is very interesting. So they wanted to ask, please can you tell us more about this and whether any other NTD is it being currently used for?
Well, I think, the mobile phone, certainly for telemedicine, I think is a great way forward. And my colleague, Rosanna Peeling, who was in charge of diagnostics at the WHO TDR program, she is very keen on having point of care tests that can be read by your mobile phone, so that message then goes somewhere centrally, so you know where the cases are occurring, for surveillance, as it were. The lower scope I've not had personal experience of using, but apparently there's an attachment. You put a special lens on the phone, and you can then detect those microfilariae in the whole blood, even without staining. I mean, the other use of a mobile phone, it's nothing to do with NTDs, but I have colleagues at the London School who are working on global eye health, and they have come up with a mobile phone which is for testing visual acuity and also, for taking photo of the eye, front or back of the eye, around the issue of telemedicine, so you can pick up diabetic retinopathy or, whatever eye disease there is. And again, that can be sent to a central area, to a specialist who can make the diagnosis and give feedback.
Excellent. I think the specific, there's similarly with telemedicine as well. There have been for TB diagnoses as well. They've been able to take X-ray images and send them across to specific institutions as well, because even getting an X-ray in LMICs is very difficult. So to have that
Yes.
interpretation really helps in the the management of infectious diseases in
Yes.
LMICs. So yes, thank you so much for that. Now the next question I have is, one of the participants have said that, dear Professor, maybe I like the WHO roadmap for NTDs. However, there is no universal healthcare coverage in Yemen currently, especially with the kind of conflict that's ongoing as well. Would there be, I guess they're kind of prompting, would there be a bit of a amendment or kind of for the roadmap in those situations?
Yes, of course, conflict makes implementation of these programs very difficult. So I think there's no simple answer. I guess you just have to take the opportunities when they arise. And I mean, the other question, so I was at a roundtable discussion actually just before I came on this call with my colleagues at the London School. We have students studying NTDs, and we had the head of NTDs at WHO there, and we were discussing, one question that came up: what about refugee camps? Are we focusing on them? And clearly, that's important too, and an opportunity to give mass treatment. But I'm afraid there's no simple answer.
You're right. Yeah, thank you. It is a very complex situation, especially with the refugee camps as well. So thank you for that.
Yes.
I do have the team asking, would Professor maybe be happy to take any verbal questions at all from the participants?
I would, but I have to go in 5 minutes. So.
No, of course, yes. No. If we have a very quick question from one of the panelists as well, let's hopefully the internet connection works. Okay, let's see. Okay, go ahead.
Hello, good afternoon. Do you hear me?
Hello, yes, I can hear you, yes.
Yes, this is Dekra, a Yemeni doctor who's responsible for NTDs in Yemen. First of all, thank you, Professor David, for your informative presentation. And I feel sorry that you are going in 5 minutes. I have a lot of questions, but I will summarize and brief you and the audience, my colleagues in Yemen, about the NTD situation in Yemen, if you allow me. I am, first of all, yeah, I am a former worker with Imperial College London, SCI. I'm sure you heard about it. And also I worked with the WHO regional office as a consultant for 1 year for NTD as well, the same as I worked with the Ministry of Health in Yemen for about 8 years, responsible for the NTDs. Okay, I will give you a brief about the situation in Yemen. First of all, I have good news, positive news. Yemen have eradicated guinea worm in 1996. So this is number one. Yemen is in the process of, or Yemen has validated LF and submitted a dossier in 2018. And I'm quite sure that there is a lot of progress after that. So also the third one, the third NTD, which is trachoma, Yemen has accomplished a very good national mapping, started from 2016 and finalized in 2018. And they achieved a good progress in regarding mass drug administration although the conflict has slowed everything now. Regarding to onchocercasis, Yemen, we are not calling it river blindness because it's not causing blindness in Yemen. It just causes dermal aspect of the disease, which is, we call it Sauda. Sauda in Arabic, Sauda means black, and because it causes black pigmentation of the skin due to excessive itching. The program is not structured well, unfortunately, because it was implemented by NGO, although we have tried a lot to coordinate between the MOH and this NGO, but also the war has distracted even this coordination. They are lacking, they were thinking about mapping onchocercasis and also doing MDA on a national scale rather than individual based at that clinic related to the NGO. Unfortunately, I have no information yet about the progress of the program, but Merck has, and Mectizan, sorry, it's Mectizan, not Merck. I think both of them are correlated. They donated a lot of ivermectin to Yemen. Third or fourth disease is dengue, leprosy, scabies, snake bites. All these are there in Yemen, and dengue is causing a big problem, an emergency problem, especially at the time of rains in the southern governorates or southern provinces. Okay, now the big one, which is I was responsible of, is schistosomiasis and soil-transmitted helminthiasis. Actually, this is a well-structured program, has started in 2010, was funded by the World Bank, and there was a strong partnership between the World Bank, WHO, Imperial College London, which I was working with, and also University of Sana'a, and the program was led by and until now, actually, it has been leading by the Ministry of Health. The percentage of, we conducted national mapping in 2014, and I was about to present that tomorrow. Unfortunately, my laptop was crashed suddenly yesterday, so I'm withdrawn from because the presentation is not there. Yes, so it was 18.3 before at the time of the mapping, and with progressive and successful MDAs national-wise, it's reduced to 3.6%. Unfortunately, that was in 2015, and we don't know how is the condition, although there is a scattered MDA still funded by the World Bank. But as you know, Yemen is under war. So the program
just in the interest of time.
Farouk, I just would like to thank the professor, and I wish that I was able to present the program of Yemen in details. Thank you very much, Professor David.
Well, you've done a fantastic job. I'm really impressed. Well done. Keep up the good work. That's amazing.
Thank you very much.
Yes, I mean that's why I love working in NTDs, because there are so many wonderful people who are doing these are running these successful programs under very difficult conditions. So well done.
And unfortunately, Yemen is neglected, and the disease is neglected, and the Ministry of Health is also neglected.
Yeah. Yes. Well, it's lovely to hear you. I'm sorry we can't meet in person. Very nice to see you, Hamza. Thank you.
That's great. Thank you so much, Professor, maybe for all your time for this. I think that's we've had enough questions for today, but thank you so much, Professor Maybe, for all your time, for an absolutely brilliant session as well. Thank you so much, sir.
It's a great pleasure. Very nice to meet you.
That's
Bye.
Great. Thank you. That's great. So thank you, everyone, for attending the session. I hope you found it as astounding as I have. I just wanted to again thank Professor Hemingway and Professor Maybe for the remarkable and exhilarating lectures today as well. And I apologize that we weren't able to go through all the questions for both speakers. However, hopefully we'll be able to do that in the future for other speakers. Now we have a 30-minute break, for which after we will reconvene for the global threats for Yemen from infection session, which is chaired by my colleague from INTERYem, Mr. Wahib Al-Azani. So we will reconvene after 30 minutes. Okay, so break for 30 minutes now. That's great. Thank you so much.
Hi, just testing my mic. Can you hear me okay?
Yes, hello. Yeah, I can hear you very nicely there. Thank you.
Okay.
4 minutes. We'll start in 4 minutes. Thank you very much for coming.
Does this mic sound any better? Which do you prefer, the first or the second?
It seems to be a lot of echo.
Yeah, because you're. Oh, really? What about the first mic?
I think.
Let me turn off my speaker. Okay, is that better?
Oh, can you try the first mic
Is that
again?
better?
Yeah, I think slightly better.
No?
I think it is. Can you hear me? I think that would do. Yeah.
Does that sound better or worse?
Can you go back to the first one just for me to compare?
Sorry, hang on. The first, okay. The first one's an open mic. This one.
All right. No, the other one. The other one. Yeah,
The,
yeah.
first, the second one, okay.
One, yeah. The
Second
second
one's a lapel mic. Hang on. So this one sounds better.
Yes, it does. It does.
Okay. I'll use this.
Great. Thank you. So you've just finished another meeting by the sound of it?
Yeah, well, I was watching the WHO meeting.
Right.
Yeah, I just finished at 5 o'clock. Just caught up on some admin stuff. So you're live streaming this to Yemen as well as the various speaker locations, is that right?
Yes, that's right. Yes.
Okay. Do you want me to use my headphones? Is that
Recorded on YouTube? So it's live streamed on YouTube and Zoom, and it's also recorded for people to watch later on the YouTube channel as well.
all right? Just see whether my earphones sound better. So I use earphones to stop some background noise. Is that better?
Yeah, I think it's better, but marginally, to be honest. I think.
Okay, if it's only marginal, I'll take them off because they feel they feel uncomfortable.
No, so if you want to keep it.
So are you going to be stepping through the slides for me? Sorry, Wahib. Are you going to step through the slides for me? Because it's much better if you step through them at your end, so I don't start talking about the next slide.
Okay, I mean, would you like, if you have the slides with you, we can give you the share screen facility and you can go through them yourself. Do you want that?
That doesn't work very well, I found, because I find that when I skip forward, there's like a 10-second delay, 5 to 10-second delay. So I start talking about the next slide when the audience is looking at the previous slide.
Right. Okay. So we haven't had this problem before.
Okay, well, let me try, because I haven't actually downloaded my slides to this computer because it's 24 megs. Hang on a sec.
Julian, I have your slides if you want me to share them for you. It's Mubashir here, by the way.
Oh, Mubashir, I think it's better if you do that because there's usually a delay from my end because it's my Wi-Fi, which is not great. So if you can step forward for me, that'd be great. Do you want to just try it before they come online? So hang on a sec, I've lost you now. Yeah, do you want to just try stepping through a few slides with me, Mubashir, just to see how it works? So if I say next slide, let's see how fast it goes. Next slide. Next slide. Yeah, next slide. Next slide. Yeah, it works quite well. So if you don't mind, Mubashir, that's kind of easier, because I find that it does start to lag, because my Wi-Fi suddenly lags every few minutes, and then I'm talking, no one's hearing me, no one's seeing anything moving.
So do you want me to share your slides, or do you want to share them yourself?
No, if you can share them, that'd be better.
Why don't you start when you are?
Is everybody online now?
Okay, yeah. I think we could start.
Okay.
So what we'll do, I'll just introduce you, Professor Tang. So, okay, 3, 2, 1. Hello, hi, everyone. Thank you very much for coming back to this second session of the Infection Conference for Yemen. The theme for this session is the global threats for Yemen from infection. We'd like to start the session by the first talk, Professor Julian Tang, who will be talking to us about COVID-19, an update on virology, diagnostics, and vaccines. So just a brief introduction on background of Professor Tang. He's a clinical virologist at the University of Leicester in the UK. His research interests are particularly focused on respiratory viruses, their epidemiology, transmission, management, treatment, prevention, and infection control, both in hospital and community settings, as well as viral infections in pregnancy. Without further ado, I'd like to give the mic to Professor Tang to tell us about COVID-19 and update on virology, diagnostics, and vaccine, and how that could be useful for Yemen.
Hello. So thanks for that introduction. So I saw the BBC documentary on Yemen, and I've read a bit about the background of Yemen since I was invited to give this talk. So I know that there are lots of problems in Yemen, not just related to COVID-19 or infectious diseases even, lots of dietary and nutritional problems, as well as obviously the ongoing conflict there. So what I plan to do is give you a background to the overall pandemic, to give you some perspective, as well as some practical advice on how to control the virus and how to treat and some of the treatment options for those patients. So next slide, please. So I understand the limitations of the facilities and the resources in Yemen. So this is really just some background information for the doctors and nurses there working with these patients. Next slide, please. So COVID-19 is the latest in a long line of pandemics. If you look at this nice perspective image, you see it goes back to the Great Plague, to bubonic plague, smallpox, some of the unknown plagues, the plague of Justinian, which might be plague or smallpox, we're not sure. And then more recently, HIV, yellow fever, and of course the recent Ebola outbreaks in West Africa and Central Africa that we've been knowing about for the past few years now. And then the Asian flu outbreaks you see in China and Southeast Asia, and of course the SARS outbreaks of 2003 and 2020 now, but also the influenza outbreaks that come every 40 years or so. So 1918, 1957, 1968, and then 2009. Next slide, please. And sorry, just back one. And the relative sizes of those balls you saw are really the impact, the number of deaths we think are associated with each of those pandemics. So you can see the Black Death, bubonic plague is one of the biggest balls, and the smallpox, Spanish flu, plague of Justinian, we're not quite sure what that might be, and then HIV/AIDS. So on that scale, COVID-19 is somewhere between HIV and malaria. So not huge numbers at the moment, but it may become seasonal going forward, like we deal with seasonal flu. Next slide, please. So this is just a more kind of anatomical background to respiratory viruses. There are lots of respiratory viruses, as you can see from the list on the top right there: rhinovirus, coronavirus, influenza, paraflu, RSV, etc. Some of them affect children more than adults. And most of the time, you don't worry too much about these. You don't have to mask for them. You don't have to socially distance or isolate or self-quarantine for these seasonal respiratory viruses. And that's because we've got lots of background immunity over many decades of exposure. And, you know, mums will pass some of the antibodies through breastfeeding and maternal in utero transfer to their babies. So we don't really notice these respiratory viruses that much that occur every season. And the respiratory viruses cause disease in the upper respiratory tract. So the common cold, laryngitis, pharyngitis, the runny nose, rhinorrhea that you see, usually not so much fever, except with influenza. But then you get the more severe disease in the lower respiratory tract, the bronchitis, bronchiolitis that you see in babies, for example, and then the adult pneumonias, pneumonitis. And other viruses can cause these more severe lower respiratory tract infections, such as chickenpox pneumonitis, measles pneumonitis, and SARS, of course, and some of the avian influenza viruses. So there are lots of viruses, and some of them may be more important than others. So this slide, you can see the diagnostic process, which you are probably very familiar with. Now you take a swab, you send it to the lab, and they run it on an automated platform typically, and you get a multiplex PCR result if you're using PCR. I grew up on things like virus culture and immunofluorescence. Those are cheaper technologies, more labor intensive, more laborious, less sensitive. But we were using those technologies until perhaps 10 years ago in some of the labs that run hospital diagnostics. And you can see from the bottom right, the viral loads can be quite high. If you take a swab from an acutely infected influenza or COVID-19 patient, you might find 10 million viruses on that single swab. This varies a lot, from 10 to 100 to 10 million, sometimes more. But typically this range is also seen with other viruses like influenza, and probably we measure it for RSV as well. Next slide, please. So here's some virologies. So the virus is called coronavirus. That's SARS coronavirus CoV-2, but also SARS coronavirus CoV-1. Looks the same, as well as the common cold viruses, coronaviruses, NR63, 229E, OC43, and Hong Kong U1. It's called coronavirus because of the spike proteins on the surface. These spike proteins, the S protein, are the focus of most of the vaccines that have been developed for COVID-19. And these spike proteins are important because they bind the virus to the cell, to the preferred host cell, via the ACE2 enzyme, which is the receptor for COVID-19, the COVID-19 virus. If the virus can't get into the cell, it can't replicate, it gets eaten up by macrophages and T cells, and it doesn't do much harm. So if you can block that receptor with antibodies induced by vaccines or even natural immunity, then you can actually stop the virus from causing infection, successful infection, and then disease. Next slide, please. And the replication cycle of coronaviruses gives you an idea of what targets you can use for actually preventing disease or preventing severe disease. So antivirals nearly all antivirals actually are just virostatic. They don't kill the virus. What they do is they stop further viral replication. That's why you have to treat patients early. If you wait too long, then there's a very high viral load in that patient. If you add an antiviral, it just stops further replication, and the body still has to deal with and clear the millions of viruses that have actually accumulated already. And this is why you see, for example, for influenza, seasonal influenza, you want to give Tamiflu within 48 hours of onset of symptoms to stop the virus load from ramping up too much. Because if it gets too high, then you start to get more severe disease. Same with HIV, same with Hep C, same with Hep B. If you want to stop severe disease, end organ disease, you want to suppress the viral load to low levels with an antiviral, and then you can let the immune system clear the virus. So here in the diagram, you can see you can actually block viral entry, viral RNA or DNA replication, viral entry into the nucleus if it's a retrovirus, for example, and then after replication, the protease inhibitors can block the protease step where those virus proteins are cut into the appropriate length to generate a new virus that then buds out. And in fact, Tamiflu works on the budding out process for influenza, where it stops the virus from leaving the cell. And something like Maravirac for HIV stops the virus from entering the cell. So there are many different steps that you can actually use antivirals for. And for COVID-19, we have remdesivir that stops the viral replication of the RNA inside the cell. And we have dexamethasone, which suppresses the immune response to the virus to prevent severe disease and organ disease when you're infected. So those two medicines can actually be working together quite synergistically to stop further viral replication, but also to suppress the immune response so you don't get severe end organ disease like ARDS. Next slide, please. So here's a bit more background about the virus. If you look at the virus, you can actually sequence the virus, draw a family tree of the virus, and you can see that all the seasonal and the more lethal coronaviruses are represented in this tree. So if you look at the top left, the 11 o'clock position, and the bottom right, the 5 o'clock position, those are the seasonal coronaviruses that don't do you much harm. They may put you in bed for a few days off work. There's a common cold virus, coronaviruses. And then at about the 4 o'clock, the 8 o'clock position and the 7 o'clock position, you have the more severe coronaviruses, SARS-CoV-1 and 2, around the 8 o'clock position. And then MERS coronavirus, which is slightly different, it tends to be in camels, reservoir in camels. It also came from bats at the 7 o'clock position. And those more severe coronaviruses can cause mortality of 5 to 30%, which is much more than the seasonal coronaviruses that we see every year. Next slide, please. So where did this virus come from? So there have been lots of investigations into this, including a WHO visit to China on this basis. And we think it came from wild animals that were naturally infected that then were caught and killed and prepared for food by humans. There's some bat soup on the right there. And the wet market on the left is where there were lots of wild animals from Wuhan in China. And there are many wet markets in China. And a lot of wild animals would be mixing in cages there, waiting for customers that wouldn't normally mix in the natural world. Next slide, please. So the way it started was that back in December 2019, there was a kind of grumbling background of pneumonia cases in China, in Wuhan. And then the Wuhan CDC sent a team in to have a look more closely at these cases. And they developed a test to check for this coronavirus. And you can see the case ascertainment suddenly shot up at the end of January. And at the end of January, that's when we started seeing imports of the virus from European ski resorts into Europe, probably from a lot of Chinese visitors who were carrying the virus inadvertently across the globe. Next slide, please. So the wet market was closed down soon after that, but you can see the active wet market image on the right, top right there, a menu for wild animal produce on the bottom left, and then an example of a market store bottom right as well, which is actually selling wet live seafood. Next slide, please. And again, it's mainly the preparation of the raw material that can cause infection, because once you cook it, clearly the virus will die. So the preparation of the raw material, just like African bushmeat or some of the other, like raw palm date juice in India, that raw material is where the virus will reside. And if you touch your face, nose, or eyes with the live virus, you can get infected. The graphic on the right is interesting. They thought it was actually snakes were a natural reservoir as well, because they eat bats, but that was later disproven. So we think the main transmission to humans has come directly from bats, or possibly from an intermediate host like the pangolin, which you'll probably see in the next few slides. And the Chinese delicacies of bat soup and bat broth on the left, actually they do this with many different animals, not just bats. Next slide, please. So the pangolin there you see at the 3 o'clock position is used in Chinese medicine. The scales are very valuable in Chinese medicine, and obviously people eat the meat as well. And again, the preparation of that meat could transmit the virus. And the reason why we think this is because when you do the sequencing of the viruses found with the pangolin, in the pangolin, the sequences lie between the human coronavirus, SARS-CoV-2, and the bat coronavirus, the original bat coronavirus that you can see in the diagram here. And that's why we think that the pangolin virus might be an intermediate virus, and the pangolin might be an intermediate host between bats and humans. Of course, you can still get direct bat to human transfer as well. It doesn't have to go through the pangolin. Next slide, please. The other thing you can do is that you can actually use these viral sequences that are dated, the sampling date, to backtrace when this virus may have occurred, emerged from a certain population, like in humans. And the team in the UK did this very early on and found that the emergence of this virus was probably around end of October, sort of mid to mid-November in 2019, which kind of fits the emergence of the Wuhan pneumonia cases. But we also think that because it's so efficiently transmissible, even from that point amongst the Chinese population, this virus must have had some time in the human population to adapt to the new host to transmit so efficiently. So in fact, the entry into the human population may have been earlier, but we're limited by the number of samples we have with dates from that time. As you can see from the top right corner, there are lots of dots after December the 31st, but very few dots before that due to sampling deficiencies, which is very common in phylogenetic analyses. Next slide, please. And what was emerging very quickly is that a lot of these cases were asymptomatic. Fewer were symptomatic, and very few actually led to severe disease and death. So this is encouraging on one hand, in that most cases were actually asymptomatic or mildly symptomatic, but it also makes it very difficult to control this virus. So if you have people who are walking around who are feeling very well and don't need to go to hospital, don't need to get tested, you don't know how big the epidemic is in that population, because you don't really see people coming forward to be tested because they feel very well. And the extreme example of this is HIV. HIV, you can have an incubation period of 5 to 10 years because people get infected, don't have any symptoms, carry on having sex with people, and they don't know they've got HIV until later on when some of their partners start to get ill, even though they may not get ill. So with a virus that spreads in a very asymptomatic manner, or a very mildly symptomatic manner, like it could mimic the common cold, for example, it's very hard to track and trace and control this virus. Next slide, please. So some of the early attempts, modeling attempts from the Imperial College team, who are now famous for this, tried to track the virus and try and calculate what the R0 was. Now, the R0 is the number of secondary cases produced by a single index case if the population is actually uniformly distributed and non-immune. Now, of course, that's not very realistic, but at the start of the pandemic, it gives you a relatively good idea of the rate of spread of this virus through a otherwise totally immune population that's never seen this virus before. And they got estimates of around 2 to 4, which sounds like quite a low number. If you imagine your chessboard analogy, if you have a doubling every 5 days, which is the incubation period for this virus, then very soon after a month, you've got 2 to the 6 copies of the virus, copies of people infected with the virus, if you like, which is quite high numbers. It goes from 2 to about 64 times the number of cases you had at the beginning, which is a huge increase. Next slide, please. And some of the early viral load studies on swabs taken from infected patients show this diverse viral load range that generally drops over time. So you can see that the highest viral loads on day, first day 0 to 3, and it drops over time to day 18 to 21. But not all the patients follow this pattern, and some patients, especially the immune-compromised patients, can actually shed live virus for up to 150, even 180 days, because they just don't clear the virus. And even if you give them the antivirals, it just stops further viral replication. It doesn't actually clear the existing virus. And these patients can stay infectious for a long time. Next slide, please. So some of our colleagues in Hong Kong, Ben Cowling and team, tried to model the dynamics of transmission of this virus with some empirical data from real cases. And they showed that, in fact, a lot of these infections occur pre-symptomatically or even asymptomatically, but before you actually diagnose that case. So a lot of the surge testing that's done in the UK, for example, trying to control the South African and the Brazilian and the Indian variant, by the time you identify a case, do a PCR, see it's positive, then isolate that case, they've probably spread the virus to, 5 to 10 other people, depending on their social contact rate before that point in time. So it's often, the horse is bolted before you've actually identified a single case that's positive. And this is why this virus is very hard to control. Next slide, please. There are some asymptomatic cases. So this nice study in the elderly population in the US showed a spectrum of asymptomatic, pre-symptomatic, and atypical, typical symptom patients. And the viral loads can be similar across the board. So you can't say that asymptomatic is necessarily shedding less virus than symptomatics. Next slide, please. Again, here, severe cases versus mild cases can shed the same amount of virus. So they can be just as infectious. Now, of course, if they're not coughing and spluttering and sneezing, just breathing and talking, that transmission rate might be lower, but we know that now that actually breathing and talking are very efficient at transmitting aerosols over a short range, that short range conversational distance, the garlic breath distance we call it. So you cannot be sure that these patients are actually not transmitting the infection, even though they're actually asymptomatic. And that's why this virus is so difficult to control. Next slide, please. So this is a nice multi-panel showing the viral loads on the left, essentially the left column, but also the emergence of antibody on the right, top right, panel D. And it shows the cessation, the stopping of live virus detection after about 8 days. Now this makes sense. If you know viruses, we know that the antibody responses don't kick in really till days 5 to 10. So there's no point using antibody testing to detect acute cases. It doesn't work because the antibodies take 5 to 10 days to appear. And if you get a negative antibody test on day 3, post-symptom onset, it doesn't mean that they're not infected. That's why antibody testing is good for seroprevalence studies, to see how much virus you've had in the population, but they're not good for acute diagnoses that you need for acute hospital admissions. Next slide, please. Now, children have been a conundrum since the beginning of the pandemic for several reasons. One, they don't get very sick. If they don't get very sick, the parents aren't going to voluntarily bring their children to the hospital to get tested. And in the UK, they didn't do any community testing for the first wave, essentially. So children weren't getting tested, so people thought they weren't getting infected. We know that's not true now. Children do get infected. In fact, they shed high viral loads for just as long or even longer than adults, as they do with other respiratory viruses, but they just don't present for testing. So you don't know whether they're infected or not. And this has been, a potential problem when schools reopen, because a lot of these children, studies have shown between 30 to 70% may be asymptomatic. They go to school to spread the virus amongst older children and the teachers, and then those children bring those viruses home to the adults at home, who are even grandparents at home, who may be more vulnerable to the infection. And we know that children are great carriers, vessels, super spreaders of respiratory viruses from RSV, paraflu, influenza, adenovirus, etc. So children are kind of a vector for the virus if you're not careful about how you monitor them. Next slide, please. And here, there's a couple of studies showing the viral loads in children. You can see that on the bottom left, asymptomatic, those with upper respiratory tract, those with lower respiratory tract, show similar viral loads. They show similar, similar viral loads for a similar duration. And it doesn't really vary whether they're symptomatic or not. And then the next slide will also show that the viral loads can be as high as adults. So if you look at the comparison between hospitalized adults and hospitalized pediatric cases in the top right panel, you can see that very similar range of viral loads. Okay, so children can carry just as much virus for just as long as adults, but they're just less symptomatic than adults. And the reason for that might be because children don't have that kind of library of antibodies and T and B cell responses that adults have built up over 20, 30 years that then may react to the virus in a way that gives them symptoms. Children are more kind of like a pristine, clean slate, and these viruses may not be able to cause, they may not cause so many symptoms because there's lack of a, well, two reasons. One, it could be a lack of an antibody immune response, but also the other theory is that they have a very hypervigilant trained immune response that actually stops the virus from causing any end organ disease as soon as they get inhaled. So there's a bit of a kind of dichotomy about the reasons why children are not that symptomatic with this virus. And it may differ between children. And of course, now we know children can get really sick. So I think the next slide. Yeah, okay, so it'll be the slide after this. So basically what it shows here is that surveillance for children has not been very good in the community because children just don't get sick, they don't get sampled. You underestimate the overall prevalence incidence of this virus in the community. Next slide, please. Okay, so I put this in for the for the clinicians out there. This is part of our local hospital protocol for treating this virus. So this shows you the kind of viral response phase in the early stage, and then the host inflammatory response phase that we think causes more severe, longer lasting disease. And this is why you need to treat with dexamethasone very early, if you're going to use it at all, and also use remdesivir, again, early, if you're going to use it at all. And you tend to only use those two interventions, as per clinical trials, for those who are oxygen dependent, so the more severely ill end of the spectrum. Now, oxygen's been a problem in places with high levels of cases, like India, for example. The oxygen dependency may be there, but they may not be getting oxygen, which is the most important intervention. If you give the dexamethasone and give remdesivir, but you can't give the oxygen, the patient's still not going to do very well. And then all the other tests down the side, again, really monitoring the response to therapy and or progression of the disease. Next slide, please. So this COVID mnemonic we developed in our hospital. Our respiratory team developed this. So caution and coagulation prevention, so wear PPE and just monitor the coagulation cascades and clotting complications. Oxygen, of course, delivered by several means. Antiviral therapy, that's mainly remdesivir now. They can also use sarilumab, tocilizumab, those IL-6 inhibitors, but they're quite expensive and quite difficult to get hold of. And then the intravenous fluids, keep the hydration optimal if you can. And then, of course, dexamethasone. And there they've included the disease-modifying drugs, the rheumatology drugs that I've just mentioned, IL-6 inhibitors, which, again, are not very easy to get hold of. So I don't know how much of this you could actually implement in Yemen, but I'm happy to share my slides if you find this useful afterwards. Next slide, please. Okay, so how can we control this virus? How do we stop its transmission? So just before I go on, I took out the earlier slides because this is mainly an adults physician population, I guess. Children can get MISC, the multi-inflammatory syndrome. Adults can still get some of this. The treatment's really some degree of immune suppression and monitoring for those complications. In children can mimic Kawasaki's disease, for example. Okay, next slide, please. So this has been a big debate, and one of my research areas is actually aerosol transmission. So we've been lobbying WHO and our own local institutions to try and enhance the awareness of aerosol transmission, the need for masking, ventilation. We're not saying don't wash your hands. We're not saying that other precautions don't work, but we think that most of the time this virus is transmitted just by talking to each other, and the masking prevents that virus from transmitting. So both source control as well as reduction of exposure through universal masking. And this has worked very well in Southeast Asian countries that reacted very early, like Taiwan, Hong Kong, Singapore to some extent, Korea. Without any modeling, they just slapped the masks on, universal masking, and the case numbers are so low. They're so low. And they actually have a normal economy in a lot of those countries now. In fact, way much earlier. In fact, Hong Kong hardly has any restrictions. Taiwan similar. China after lockdown is similar. South Korea after the little surge and first wave again is doing very well. A lot of the Western countries didn't do this because they're not used to masking. They denied the effect of masking, And the numbers of the virus just surged throughout the whole of Europe and North America and South America as well. Next slide, please. So this has now been updated. So the CDC website now and the WHO website have much stronger message now, saying that aerosols are the main transmission route. Just breathing and talking can produce aerosols that can be inhaled by others and spread the virus. So the masking message is more important. The ventilation message, clean air, filtered air is more important. And the social distancing, because distance protects you against all forms of transmission, whether it's touch or aerosol. And droplets are aerosols if you can inhale them. If they are too big to land on the floor, they're not aerosols, they just fall to the floor and don't bother anybody. And they may form a source for contact transmission, but we don't think that is a major way this virus transmits. So a lot of the surface cleaning that people are doing are probably mostly a waste of resources, but people feel better doing it. So, you know, if they want to do it, that's okay. But if you're talking to a stranger that you don't actually shake hands with or hug or kiss, the way this virus transmits is by aerosol through that shared airspace between them. Next slide, please. So I developed this imaging system in Singapore, which gives you a real-time shadow graph, slurring image of your breath that can cross between two different people when they're talking. Next slide, please. And here you look at so-called differences in the diffraction patterns of the air. The warm air leaves your mouth and nose and hits the cold air in the space between the two of you, and you see that shadow of that different diffraction index when the light passes through the warm air into the cold air in the environment. You can see the way the breath travels. As you can see there on the left, that's me just talking and breathing at the same time, and producing that little plume of shadow you can see there, which could be carrying virus as well. Next slide, please. So this paper caused a lot of stir. They thought this paper implied aerosol transmission because of high survivability of the virus for 3 hours in the air, but also on surfaces like plastic and cardboard and steel. And the idea is that these are lab experiments done in optimal conditions for enhancing virus survival. If you have virus landing on a steel car hood, for example, in the sunlight outside, it's not going to survive for 3 days. It'll get killed by the weather, by the sunlight, etc., etc. So these are very lab-based estimates, but it does show that in indoor environments, for example, airborne virus can survive for several hours. Viruses on surfaces like plastic and steel surfaces in wards, it can survive for quite a while if you touch them, but the infectious dose will be very low if you touch them. That seems to be the main issue about why contact transmission doesn't really transmit this virus. But if you have an aerosol that you inhale, we think the infectious dose might be just 300 to 500 viruses if you're non-immune. You can inhale 300 to 500 viruses just in one breath if you're talking to a highly infectious acute case of COVID. Next slide, please. And this is a nice study from actually pre-COVID pandemic, 2019, showing the number and size of particles produced by the nose and mouth during breathing and talking. And you produce even more aerosols by talking louder, which makes sense because you're forcing more air through your mouth when you talk louder, when you shout, when you sing. And these aerosols spread out into the immediate vicinity, and if they're carrying virus, a lot of people can actually inhale that virus. So I'm thinking that, if you get friends to sing happy birthday to you around the birthday cake, and you're sitting down and they're standing around you, you have a lot of exposure from all of them singing happy birthday to you. So it may not be a very happy birthday for you if you get infected afterwards. Next slide, please. Again, this is an early study showing that if you cough, well, this is actually a sneeze. Sneezing produces large amounts of droplets of different sizes, producing an aerosol that can travel up to 7 to 8 meters. Even though you can't see it, those particles in that aerosol cloud can transmit the virus over long distances, much more than 2 meters, as you can see there. Now, of course, we don't cough and sneeze all the time, but we do breathe and talk a lot of the time. And 2 meter distance is probably much more, it has been shown to be much more effective than 1 meter if you're just breathing and talking quietly as a source of the virus. Next slide, please. And there's another study we did in Finland showing that the use of nebulizers in a patient lying on the bed, a patient with perhaps a respiratory virus infection and or asthma, the nebulizer can actually shed the virus, can spread the virus around up to 2 meters. And we found different levels of virus from this mannequin study using a home nebulizer with a simulated aerosol actually consisting of the vaccine virus, the Fluenz pediatric live attenuated vaccine virus as a tracer. And this is probably the same with COVID. You probably see some live virus shed during nebulizer use as well. Next slide, please. And again, on airplanes, the ventilation is very good. It's very quick. It's really 30 air changes per hour. So the air is actually replaced every 2 to 3 minutes in the plane fuselage. But the risk is mainly from the person sitting right beside you. So if you're talking to somebody right beside you, that virus has already moved across that space before the ventilation can take it away. So if you wear the mask on the plane in a high prevalence situation of the virus, then that will also add to your protection, as well as the plane ventilation system that's always swirling around you. And this is the difference between long range and short range aerosol transmission. And this is a concept that's been very hard to get across to people, actually, particularly in the infection controls teams, where they think that everything's droplets. In fact, droplets are now really things that fall to the ground and the gravity, and aerosols are anything you can inhale, whatever the size is going to be, however far it's actually traveled to reach your nose or mouth, it's still an aerosol. So if you wear the mask and you mask a source, it reduces the overall aerosol population in that airspace before it can transmit the virus to anybody. Next slide, please. And they found live virus in the air, okay? But don't forget, when you sample the air, the source has probably gone away and may have gone away for several minutes or even hours. So what lingers in the air does not really represent the true concentration of virus that you get from standing in front of somebody and talking to them. And that's why air sampling studies are actually quite misleading, because a lot of them find no virus or very little virus or non-viable virus, but the virus could have been there for hours, you don't know. And the RNA could be wafting around for hours, but in fact the viability is gone, but it doesn't represent the risk of standing in front of somebody with COVID, talking to them in real time within a meter. And they're producing fresh virus all the time, every time they breathe out. So whilst these air sampling studies in the environment, in the wards are quite useful to see what the minimum, at least that amount of virus is, it doesn't represent the amount of virus you get from somebody standing in front of you, talking to you during a coffee break, and breathing in your face whilst they're talking and listening to you. Next slide, please. Similarly, the air sample is very important. The air sample produces a very strong airflow that shears the virus, it shreds the virus envelope and makes it less infectious. Whereas if you're breathing and talking with somebody, your inhalation breath is actually very low velocity, less than 1 to 2 meters per second. The virus is not sheared, not damaged. You may inhale most of the live virus from your friend who's talking to you. And if any of you actually smelt your friend's breath, they may have had garlic or curry or alcohol for lunch. If you can inhale that air, you're actually inhaling any virus that might be carried in that air space as well. And this air sampler is a very gentle air sampler, and it picked up live virus. But even that would really underestimate your exposure because the source could be anywhere. Okay, even though the patient's there, the patient can breathe, for an hour, but the air goes everywhere, and the sampler is just in one place. Okay, so it really underestimates the amount of virus that could be produced by an infectious source, even if they're quite nearby, because of that kind of dissemination in the 3D space. So these air sampling studies, whilst they're useful, they almost certainly underestimate the risk of aerosol transmission over time and distance. Next slide, please. So the controls for airborne and other, well, mainly airborne infection are really showing that PPE is the lowest form of interventions. It's low because it depends on human behavior. People don't wear the mask properly, they don't gown up properly, they don't wear the gloves properly, or they touch their face. If you can introduce efficient ventilation, which is the yellow bit, the engineering controls, that doesn't need any human intervention. We do need to monitor it for checking the effectiveness, but that's much more effective than having people wear PPE all the time, because they just get fed up, they get irritated by the skin contact, they don't wear them properly. and it's hard to breathe. So what we're advocating is to improve the ventilation across indoor spaces, both recreational and office and schools and hospitals, to try and actually reduce the overall airborne concentration of the virus and everything else to help people stay more safe. And ventilation can also reduce things like allergens, reduce hay fever, reduce sick building syndrome, absenteeism, reduce the other respiratory viruses, not just COVID. So it's really a catch-all for a improved, safer indoor area. And of course, it can be on demand. So if there's nobody in the room, it can turn off, just like the lights do in some buildings. And when it's full, you can ramp up to a higher ventilation level, and you can even just open the windows. So a lot of these old buildings just have windows, like school classrooms, open the windows in summer, and that ventilation can be very powerful to replace all the airborne virus that's being produced, breathed out by all these children and all office workers who are occupying that space. Next slide, please. So masking. Now, I mentioned masking several times already, and the Western countries refused to mask. They didn't believe in it. They thought it was, just a waste of time. But in fact, Southeast Asian countries that use the masks and have been using them since 2003, since the original SARS outbreak, and then again, with MERS and avian flu, etc., all these different threats, they're very used to wearing the mask, and even children wear masks very well. I think there's a photo later on of children wearing masks. So the masks reduce the source exposure. Even a surgical mask can reduce the source exposure by 4 to 6 times. So if you have 1,000 viruses being breathed out, if you wear a surgical mask, you can reduce that virus load to about 250. And then the person wearing the mask on the other side, who's being exposed, that mask can reduce that exposure by, again, 4 to 6 times, or 2 to 4 times. So it can reduce that 250 down by another 4-fold, down to like 60 viruses. So instead of being exposed to 1,000 viruses, if you're talking to somebody and you're both masked, that virus exposure can reduce down to just 60 viruses, which is much safer than being faced with 1,000 viruses coming towards you if you're unmasked and sharing the same airspace. And this is why we think the virus has been so poorly controlled in the West, because people didn't believe this, they deny this, they argue about this. And by that time, you got 100,000 people dead in the UK and more in Europe and even more in South and North America. I think Asia has probably the combined death rate of a small Western country, maybe not even that. Next slide, please. So some of the studies have shown that if you put a mask on a mannequin head and you spray aerosol influenza or just spray particles towards it, the probe inside the mask picks up very little of the spray, and that shows the protection from the outside. And if you do the reverse, you can actually see what they breathe out, and then how much the mask stops the exposure to the outside. So this one actually stops the wearer from getting exposed. And you can see at the bottom right, there's FFP3, FFP2, FFP1, and then surgical masks, which are less effective by quite a way, but better than nothing. Okay, so the protection factor drops from like 100 plus down to like maybe 5 to 6, but still that's better than nothing. Okay, so if you think about it, that you're reducing the aerosol viral load of 1,000 just by 4-fold, like it shows in the diagram, you're reducing that exposure by 1,000 down to 250. And if that person's masked as well, it's already down to 250 by the time it leaves that person's airspace, goes around the mask, for example, because the mask's not airtight around the face. It comes towards you, it can reduce, and your mask will reduce that amount by another 4-fold, so it could be down to 60 viruses. That reduces that transmission risk to you. Okay, and vice versa, because it works both ways if you have universal masking. Next slide, please. So this slide from Don Milton, one of my collaborators, shows the opposite. So that if you have people breathing into a cone to capture all the viruses, when you put a surgical mask on them, it can reduce the amount of virus significantly by about 2 to 4-fold. Okay, so you can see on the diagram on the left, if you wear a mask, it stops the larger particles completely, the coarse particles, he calls them here. And then the fine particles, it also reduces them by about 2 to 4-fold. Again, better than nothing. But if you have both people wearing masks, you reduce that exposure both directions to both of you, because the virus is trapped in the mask. That's what the mask is supposed to do. Okay, so the cheapest, most versatile intervention, if you can't get indoor ventilation working very well, or you can't open the windows, if you wear the mask, it protects others from you because it reduces your outward exposure, but it also protects you from others if the virus is coming towards you. Okay, so for protection, this is the most important message I should give you. And the masks do work. They work better if everyone's wearing them. And the masks are cheap, and you can actually sterilize them yourself. Don't soak them in water. But what I do is I spray my hand rub in a spray on the inside and out the mask, and you let it dry for 1 to 2 days, and you can use it again. So I've got like 4 or 5 masks that I just rotate on a regular basis if I need to wear them. And in fact, I've only thrown, I think, 2 masks away in this whole pandemic because I'm lab-based. I don't need to see patients. I only wear the mask if there's a colleague in the same room as me, and I have my own room, so I don't need to wear the mask very often. But when I go to the supermarket, I do the same thing. Okay, so I've had COVID in the first wave, probably from a family member. I got natural immunity. I've had the first dose of the AZ vaccine. I get the second dose later on this month. But I think the natural immunity plus the vaccine immunity will work well to prevent severe disease in you if you can get the vaccine. I know there are lots of supply problems in Africa with COVAX not working very well at the moment and problems with supply globally. But the principles there, if you can get vaccine, get the vaccine. If you can't, get the mask, wear a mask until you get vaccinated. And ideally, once you get vaccinated, you still wear the mask. It gives you further protection. Next slide, please. So these are some innovative masks in the early pandemic. In fact, the top right, the bra is actually from 2003. And then the bottom left is from 2020 in Hong Kong. They wore a mask, but they wear the bottle for eye protection. And in fact, he's got glasses on as well. But the bottle's actually quite useful because those water cooler bottles have a little chimney at the top so you don't get fogged up. And they just cut off the bottom, put it over the head. And it actually works very well, but it looks a bit strange. Next slide, please. And in children, so children in Southeast Asia are used to masks. They've been using masks for, almost 17 years, almost 20 years since 2003, SARS, but also they wear masks to reduce pollution exposure in Southeast Asia because they have been, coal burning countries and also forest burning like from Indonesia on a regular basis. So the Southeast Asian children are used to wearing masks. And you look at Thailand, not only are they wearing masks, they have a little cubicle around them that stops any aerosols coming in from around the sides because the whole desk is protected. And then in Japan, they use the visor. The visor is not very effective. But, children from primary school and secondary school can wear the masks. In Taiwan, they have an enclosure as well. And in China below, you can see that it's 1 dinner table per pupil. And they have a mask in between the eating stages. So children can wear masks. Certainly in the West, they think that they can't, which I think is something that the adults impose on the kids. I think if you teach the kids, they can do it. Next slide, please. So vaccines. Now, this is the list of the UK vaccines. Moderna, so the COVAX charity will try and distribute probably mostly the AstraZeneca vaccine because it's the cheapest one. It's sold at cost price. And the manufacturing is actually fairly well ramped up for that. Some of the other vaccines cost more and are more limited, but the AstraZeneca vaccine has had its own problems, all the kind of regulatory issues, the rather heterogeneous clinical trials that make the US reluctant to license it, the clotting complication issues, the 1-dose versus 2-dose regimen, and the long interval between the first and second dose. But in fact, the real world data suggests it works quite well against most variants, except for the South African variant. We think it works against the B117 Kent variant very well. That's now widespread. Also, the new Indian variant, the 617.2, it may work quite well against that because it doesn't have the 484Q mutation. That seems to be a problem. So you may well be offered the AstraZeneca vaccine in South Africa, in Yemen, in Southern Africa, and the rest of that continent as well. So I think that the AstraZeneca vaccine is better than nothing. If you can even get 1 dose, I try and get that 1 dose. And we found that if you take paracetamol, if you get paracetamol, it can reduce the side effects even more. And that was in the clinical trial arm as well. The other vaccines, the Chinese Sinovac, Valneva, whole virus vaccines, Chinese Sinovac hasn't been working very well in some of the countries that have been using it, like Chile, for example. Not quite sure why that is, because whole viruses tend to be quite immunogenic. They should work quite well. The mRNA vaccines, the Russian and the Moderna, Pfizer, they're doing better against the various variants, but they cost more and they have some storage issues, although those are improving now. But still, you know, they have -20, not -70 anymore, but some of them can be actually lyophilized, that the Russian Sputnik 5 can be sold as a powder that you make up yourself. But that still needs to be stored up at 4 degrees just in the fridge. So there's a wide variety of vaccines, but I know that the COVAX system will be limited to what they can actually supply to Yemen and other low middle income countries. Next slide, please. So a bit more theory. So the Novavax GSK Sanofi vaccines are basically based on the influenza vaccine design. It's a subunit virus vaccine, subunit virus protein, so a single protein, the S protein that's grown in, say, bacterial culture or yeast culture, and purified and then put into the vaccine as a single virus protein vaccine with various adjuvants and excipients. The adenovirus vector vaccines, the ones that cause some of the clotting issues, they use the adenovirus vector and then they insert the S protein gene into that from the virus. And then when it's injected, that S protein gene is then translated into S protein that then induces an immune response in that vaccine recipient. The reason why we think that the clotting issues appear is because when you get an inflammatory response to that vaccine, it seems to trigger what we call a heparin-induced thrombocytopenia type mechanism to then create antibodies to platelet factor 4 that can cause clotting issues in those vaccine recipients. That's relatively rare, 1 in 100,000 to 1 in a million, depending on which study you look at. And the vaccine can still offer you more protection against the natural infection, including clotting complications, than if you don't take it. So I would really urge you to take the vaccine. The Russian Sputnik 5 vaccine hasn't declared any issues with clotting, but a lot of that data is not very open. And the Johnson & Johnson, the Janssen vaccine, and AstraZeneca have been investigated, and there is a very low risk, which is why in the UK they're offering a different vaccine, one of the mRNA vaccines, to the under 40s, because that risk benefit ratio starts to get more kind of equivocal as you get younger. So you want to avoid giving a vaccine that causes clotting problems in the younger population, because the risk of the COVID infection is much lower as well in those younger populations for severe disease and death. And of course, the mRNA vaccines, they actually have mRNA coding for S protein wrapped in a so-called lipid nanoparticles that are injected into your shoulder as well, into your deltoid muscle. And those trigger an immune response when that S protein is translated from the mRNA in those muscle cells and dendritic cells that then induce that vaccine immune response that then protects against the S protein of that virus. Now, of course, the variants can escape some of these protective effects because the original vaccines are made to the original Wuhan virus S protein. But a lot of these new variants have mutated in the S protein gene, and they don't quite match the vaccine antibody induced by these vaccines that were designed on the older Wuhan virus. So some of these companies like AstraZeneca are thinking about redesigning the vaccine and giving a third booster shot that will cover the South African variant mutations, for example, in the S protein. And the 484K, 484Q mutation may also be covered, and that's also present in the Brazilian variant and the Indian variant as well. But of course, you can't make a vaccine that covers every single variant. It's just you can't keep up with them because they appear before the vaccine's actually been manufactured. So like influenza every year, you're always trying to catch up with the virus. Next slide, please. More complicated diagram. Now this just shows you the mRNA vaccines that we're using at the moment are so-called non-replicating mRNA vaccines. In the top, Pfizer, Moderna, and the Russian Sputnik 5. Sorry, I think the new German CureVac is also non-replicating. But you can get self-replicating mRNA vaccines. Arcturus in Singapore and the Imperial College team in the UK are looking at self-replicating mRNA vaccines that can maybe just give you protection with one dose, because the viruses, the viral proteins are replicated inside you by that self-replicating mRNA vaccine. The non-self-replicating vaccines, the top ones, Pfizer and Moderna, you have to give 2 doses and maybe a third one as the vaccine immunity wanes over time. Next slide, please. And this is just a nice cartoon showing that what happens when the vaccine is injected into your deltoid, your muscle cell, those liquid nanoparticles release the mRNA that's coded by the cell itself to produce S protein, and that's expressed on the surface of the cell, that's recognized as foreign by your immune system, and then that whole antibody B and T cell response kicks in, and it develops a long-lasting immune response that would hopefully protect you for at least 6 to 12 months from that original virus that it was designed against. So of course, some of the variants now have been tested against this Moderna and Pfizer vaccine, and it has some cross protection against the other variants. So they compared it against the South African, Brazilian, and I think the Californian variant, and even the original mRNA vaccine designed for the Wuhan virus seems to give you some cross protection against these South African, Brazilian, Californian variants, which is very interesting because the US, they only have Moderna and Pfizer for most of the time they've been vaccinating, and the Californian variants and South African and Brazilian variants, there's some protection against those by those mRNA vaccines, to the extent that in fact Pfizer's recently declared that they're not going to update their vaccine, unlike AstraZeneca, who plan to update the vaccine to cover the South African variant. So those mRNA vaccines seem to be the creme de la creme of the vaccines, but you're also paying for them. Next slide, please. So the Chinese Sinovac vaccine is a whole inactivated virus vaccine. It doesn't seem to be doing very well. So the efficacy is more closer to 50 to 60% rather than 80 to 90% that you see in the top left there. Brazil have downgraded that efficacy claim because they did some further testing. And in Chile, most of the vaccines, actually the Chinese Sinovac vaccine, and they have an ongoing surge in cases there. So it may not be as efficacious as we would hope. And now the Chinese team are trying to mix and match the vaccines like the UK is, trying to optimize that protection with the different vaccines that they have. And that also includes the CanSino vaccine, which is an adenovirus vector vaccine like the AstraZeneca and the Russian Sputnik 5 vaccines as well. And now I think they're developing an mRNA vaccine in collaboration with Pfizer, with BioNTech. Next slide, please. So this just summarizes the kind of vaccine-induced mechanisms. So some of the vaccines induce a stronger T cell response, which you can't really measure, except in the vaccine trials routinely. And some induce a stronger B cell response. So, for example, there was some discussion about mixing the Pfizer and the AstraZeneca because we think the Pfizer vaccine induces a stronger T cell response, whereas the AstraZeneca induces a stronger B cell response, the antibody response that you can measure. So there have been some trials ongoing for that, and the most recent update from yesterday was that if you give the two vaccines separately as a first dose, you get kind of the same spectrum of adverse effects with each vaccine first dose as you would expect if you're just going to give a first and second dose of the same vaccine, which is not that surprising because the adverse effects, the body will recognize them in different ways, so you won't get that kind of second dose recognition that you would get with a so-called homologous prime boost response with the same vaccine versus this heterologous prime boost response with different vaccines. And they're looking at two different intervals, one 28-day interval, one 84-day interval, the 12-week interval, to see which interval gives the most optimum protection depending on which vaccine is given first. Next slide, please. Yeah, and just more of the same here, comparing T and B cell responses. But all the vaccines give you some degree of B and T cell response, so that's not a problem. The wholly inactivated virus vaccines might give you, you might think would give you more of a broad spectrum response because it gives you an antibody response to all the different virus proteins versus just the S protein vaccines. But the whole virus vaccine responses to the Chinese Sinovac Sinopharm vaccines have been less than hoped for. but there could be some other issues about the different excipients or possible adjuvants they might be using or might not have in the vaccine at the moment. Next slide, please. So this is the idea between 2 doses. Why do you need 2 doses? Well, with the non-replicating adenovirus vaccines and the subunit protein vaccines and the whole virus inactivating vaccines, you want to prime the system first, just like natural infection primes the system. And then after a certain interval, once that immune response is established, you give the booster dose, which is like exposing the person to the virus later on. And that boosting, that exposure boosts the original primed immune response. So those of you who know something about immunology, the primary immune response, you see IgM first and then IgG slower coming up after about 5 to 10 days. But once it's primed, when you get a re-exposure later on, the secondary immune response is a rapid IgG response with a slow or non-existent IgM response. And what you want is the IgG response, because the IgG response is very good at neutralizing the virus and opsonizing the virus for other immune system cells to actually eat the virus in that opsonized antibody antigen complex. So that's why you have 2 doses. Now you might say, well, why is the flu vaccine just 1 dose? Well, the reason why it's 1 dose, I suspect, is because people won't come up for a second dose, and 1 dose is not very effective. The influenza virus vaccine has been quoted as being like 0 to 70% effective. And if you look at most of the studies, it's around 50 to 60% effective. So like the AstraZeneca single dose, well, even 2 dose, full dose response in the clinical trials. So not great, but it's still better than nothing, particularly if you're in the vulnerable group that we try and encourage to be vaccinated every year in the seasonal flu with the flu vaccine. Next dose, please. Next slide, please. So this is just a summary of the different amounts of virus COVID-19 vaccines that the UK has purchased. Just a lot of them. Okay, more than enough, full dose per individual. And these are all designed to the old Wuhan virus strain. Like I said, if there's a significant change in the antigenicity, as you can see with some of the variants, these vaccines may be less effective against them, but they will probably present severe disease and death. So, excuse me, even if they go via COVAX to other countries, having those vaccines is still useful because that broader spectrum of protection will protect you from severe disease and death, even though you might get some symptoms which would probably be mild to moderate if you get exposed to a different COVID-19 variant, for example, SARS-CoV-2 variant. So they're still useful, better than no vaccine at all. Even 1 dose is useful, better than no vaccine at all. Next slide, please. So these are the cost slides. Now, if you get your vaccines through COVAX, then it'll be free. But the relative costs are quite significantly different. So look at the Oxford AstraZeneca. Its cost price about £3. In fact, I think it's probably about £1.50. And then it's obviously delivery and transport, which will cost something. Compare that to, say, the Moderna, it's 8 times more expensive. Yes, the efficacy is higher for Moderna and Pfizer-BioNTech, but you're paying 5 to 8 times more for them. And in fact, in the real world populations, the AstraZeneca is still quite effective, even though it's even 1 dose, even though it's actually much cheaper. And that's why COVAX and Gavi are really supported by the AstraZeneca vaccine at the moment. Next slide, please. Now, with all these vaccine trials, if you read the papers, you've got to figure out what is the endpoint of the vaccine trial. Is it severe disease and death? Or is it death? Is it hospitalization? Or is it PCR confirmed infection? Regardless of whether you're symptomatic or asymptomatic. So if you compare these different endpoints, they're not the same for all these different trials. Most of the trials will have PCR confirmed infection with or without symptoms as their endpoint. And then severe disease and death is like a secondary endpoint as such. But in fact, the vaccines are not designed to actually prevent transmission. So preventing transmission is something that's not usually monitored, with Hep B or yellow fever or Hep A or flu vaccines. This has become an issue in the COVID-19 pandemic because the problem with onward transmission is that you might transmit to somebody who's vulnerable, not vaccinated, and then they may get severe disease and end up in hospital. But vaccines tend not to be designed to prevent transmission. If they do, that's great, but the vaccine takes 3 to 4 weeks to actually develop a full immune response. In that time, if you get exposed, you may be incubating the virus at the same time that you're developing an immune response to the vaccine, as well as a natural immune response to the virus as well. And that takes some time to develop, and the virus could replicate to a certain extent and be transmitted on before that vaccine or natural immune response kicks in. So you may well get some onward transmission at low level whilst that immune response is actually developing. Okay, so the vaccine is not designed to prevent transmission, and if you get at least 2 to 4 weeks between vaccination and exposure, you'll be better protected after that. Next slide, please. Right, so for those statisticians amongst you, you can do this calculation yourself. It's simply a relative risk calculation where you put, number of vaccinated and exposed, not vaccinated and exposed into a 2x2 matrix, and you can do the calculation very easily. This is all online if you're interested to do this. And that's where you get these magic numbers of 91% efficacy, 94% efficacy, 61% efficacy, etc. That's quoted in the press a lot. Next slide, please. And this is just some data from one of the earlier AstraZeneca vaccine trials showing that you do get some measurable protection after 1 dose. So you can see that the first dose on the right, you get some protection. The blue is the incidence if you're not vaccinated with 1 dose, and the red is the incidence if you're vaccinated with 1 dose. So that gap shows some protection. And obviously that gap increases over time, or is maintained over time if you have 2 doses. So there is some data for first dose versus 2 dose regimens. And that's why the UK and now other countries are going for a 1 dose for more people rather than 2 doses for half that number of people to protect them from severe disease and death from COVID-19. Next slide, please. Lots of variants now. These are the early ones. This is the B117 Kent variant. Because the vaccines target the S protein, they're very sensitive to S protein mutations. So most of the mutations you see in the press are really related to the S protein mutations. And the S protein has a binding site, the kind of receptor binding site, the RBD, and then they have the rest of the S protein which is not involved in binding. So here you see for the Kent variant, 501Y is kind of in the binding site, whereas the 6970 deletion is not in the binding site. And so you have 2 areas where you might get increased transmissibility and more vaccine resistance. So the 501Y we think enhances transmissibility, and the 6970-del we also think enhances transmissibility. There's no 484K in the original Kent variant. So in fact, the Kent variant, the original one, is still well protected against by the AstraZeneca and Pfizer vaccines. One of the derivatives of this has evolved, the Bristol variant, which contains a 484K, and we anticipate that will be less well protected against by the current vaccines. Next slide, please. So here you can see the receptor binding domain. If you look at the bottom left picture, the receptor binding domain is that kind of little blob on the top that binds to the cell, the human cell that it wants to enter. And then the other parts of the S protein, like the internal domain, you can see it lower down that say the 2 o'clock position, they're not within the binding site, but conformational changes of those other areas may actually affect how the virus behaves as well, including enhanced transmissibility, and also PCR misses. So that 6970-del is responsible for what we call the S gene dropout in the UK, based on one particular assay called the TACPath assay that targets N, E, and S, 3 targets, but the S target doesn't come up if it's a variant, a Kent variant like this is. Other assays don't target the S protein at all because some of them know that the S protein is going to mutate first. So they target the N protein, an internal protein, and the E protein, a more stable envelope protein. And then you don't get this PCR dropout or miss get a false negative when you're testing a sample. And you can see the South African variant there that's mentioned has a 484K mutation as well as the K417, which may contribute to more significant vaccine escape. So the next slide I think shows that. Yeah, so the South African variant spread very quickly throughout regions of South Africa, just like the Kent variant spread through the UK. If you look at the ratios of non-variant versus variant cases, you find that the variant cases quickly dominate the number of infections in the population, which is how they picked up the Kent variant as well in the UK. And the 484K mutation is also found in the Brazilian variant, and the 484Q mutation in the same location, but just different amino acid, is found in the Indian lineage variant 1 and 3. It's not found in the variant 2 version of the Indian variant. And the 417N is 417T in the Brazilian variant, may also contribute to the more enhanced transmissibility we see in Brazil as well. So these S protein mutations can have fairly major impacts on your vaccine efficacy and the amount of protection that you get from the vaccines, but that's to PCR confirmed infection. So you may get infected, but we think that the broader protection offered by these vaccines will prevent more severe disease and death, okay, even with different variants co-circulating. Next slide, please. So I think this is the last slide. So the question is whether this virus will go away, which we don't think it will because it's too widespread, whether it's going to become sporadic, like avian flu or like MERS that comes and goes and affects certain individuals and certain populations now and then. Maybe not. We think it's going to be more seasonal like flu, the last option. So seasonal flu vaccine, seasonal COVID-19 vaccine, it may well become a more kind of stable oscillation between winter flu COVID season and summer, hopefully summer COVID-free season. Okay, so then you have some normality between during spring and summer, and then vaccine related protection required during the autumn and winter. I think that's the last slide. Okay, so I don't know whether there's any questions or there's any time for questions, but thank you for listening.
Thank you, Professor Tang, for this amazing lecture, very detailed throughout. And I thought I knew some about COVID vaccine, but and also transmission and treatments. But I think there's so much I've learned today, and I'm sure everybody else will feel the same way. We are actually running quite behind, and but I think there are a couple of questions that I would like to. We won't go through all of them because they all will take time, but there are some that I think would be very, very beneficial for the people in Yemen. So if you don't mind answering some of these, and I hope the following speakers won't mind if we slightly start their sessions a little bit later. So Professor Tang, one of the questions is the science of virology in Yemen does not exist. That's one of the people asking that. What would you think would be the best strategy for establishing such a thing?
Okay, so you need the virologist there. So you need someone who's experienced in lab virology to set up the lab. I saw some of the facilities donated by MSF. So the biosafety cabinet is key. The PPE is key. The pipettes are key things. If you want to do antibodies are easier than PCR, but antibodies don't tell you what's happening acutely. So some of the lateral flow tests you can do quite easily. You can do it safer in the BST, biosafety cabinet. There are single-use, there are kind of fire and forget platforms like the CEFID platform for PCR testing, and we use that for confirmation as well as rapid screening in our hospital as well. And those of you who know the CEFID, it's just a box that you actually have a cartridge. You load the sample like a swab collected in transport medium. You just load a bit of that sample with a pipette. You put it into the box, you close the cover, and you wait for about 30 to 40 minutes, and the result will pop out. The problem is that it's only a single sample machine. So you can get multiple channels, like 4, 8, 32 channels, 16 channels, but they cost more money. And I know that CEFID is actually covered by WHO sponsorship for some situations to try and enhance rapid diagnostic testing in certain facilities. So ideally what you want is a lateral flow test, which is much cheaper, about maybe £5 to £10 a test. And then the PCR machine, like the CEFID, for example, which is more like 30 to 40 pounds a test. The lateral flow test takes you 10 to 20 minutes. The PCR test takes you 30 to 40 minutes. And you have a team that can do this testing in a competent, safe way. In fact, we have this in our hospital as well. We have the main lab, then we have a satellite lab that covers rapid testing off-site in a small lab with a biosafety cabinet as well. I think those standalone platforms are the easiest for the teams to use in Yemen. If you can get those, and maybe via WHO. So the CEFID platform, you get like a 2 or 4 channel one, is very useful. And then the lateral flow tests for both antibodies and antigen testing could be a backup, could be a frontline screen for that, with a CEFID as a backup confirmation test.
Great, thank you very much for that. And I would like to invite anybody in Yemen who would like to pursue this further to get in touch with us, and we can link them with the appropriate channels. Another question here is, with especially with having variants popping in there and then everywhere, there is a good chance that a variant in Yemen might have happened. Now, the vaccine which will be distributed in Yemen will be the AstraZeneca. What would be the best way to see whether there is a variant in Yemen and whether the AstraZeneca would work against this variant or not?
Okay, so what you really need is a sequencing service, and that's even more complicated to set up, unless you can send samples away. But if you send them away, it takes several weeks to come back. You kind of already missed the boat. There is a new test coming out that we're trialing in our hospital, which actually is a PCR test that targets different variants, so-called a genotyping test. We have these for things like hepatitis C as well. If you can get a PCR system set up and apply that genotyping test to different samples, you might get an idea of which variants are circulating. So at the moment, that kit only covers the Kent variant and the Brazilian and the South African variants. It doesn't yet cover the Indian variant or any Yemen variant. And to actually discover if you have a unique Yemen variant, you need to then send your samples from your Yemenese patients to a lab that can collaborate with you and do the sequencing for you, which they'll almost certainly do for free. I think the UK team, the COG UK team, are working with WHO to try and offer this service to a lot of low and middle income countries now.
Oh, great, thank you. So really, it's a lengthy process and it requires a lot of resources. So the best course of action is...
A lot of resources locally, or if you send it away, it needs time for that result to come back to you.
Yeah. So really, the
Can I
best
just answer this question about the mask?
Yes, that will be next. But I just want to say the best course of action is to take the vaccine right now, and then work on this process simultaneously. Yes, the question about the mask is very important. So if you may, that would be great.
Yeah, so you need to be a bit careful about what you clean the mask with. Now, alcohol is good to kill the virus because it's a lipid envelope virus. But when you add alcohol to the mask, you might damage some of the fibers. Okay, so you need to experiment with this because alcohol will dry on its own for sure. But the alcohol may damage some of the fibers in the mask and reduce their trapping efficiency. What I do is I use a water-based disinfectant because your breath is water-based. And then I spray it lightly on both the inside and the outside, and then I leave it. And in fact, if you hang your mask in the sun, because you have lots of sun in Yemen, you may not even need to do that. If you hang the mask in the sun, like on a washing line, the ultraviolet light from the sun will kill the virus on both the inside and the outside. And you won't lose the integrity of the virus through any wetting with alcohol or water or other things. So I think if you can rotate the mask, like if you have 2 masks, you wear one on alternate days, and you have a washing line at the back of the hospital, and you hang the masks up, and you label them for different healthcare workers, and they can rotate the mask on a 1 to 2 day basis. So for example, you might have someone called Mohammed wearing a mask on Monday, and he wears a different mask on Tuesday, whilst the Monday mask is drying in the sun. Okay, and then the Monday mask should be safe to wear, say, on the Wednesday. Now, of course, this is very ballpark. You really have to swab the mask and see if you kill everything on the mask. But I think the sunlight, we know, will kill the virus after just a few hours. There's several papers looking at this with simulated sunlight. I can send those to you via Mubashir if you want to have a look at this. But I think sunlight is a resource you do have there, more than even in the UK. And sunlight is very effective in killing lipid envelope viruses.
Thank you. And just following on that, in terms of fabric or material for the mask, is there any particular material that has been shown to be better at, for example, preventing transmission, or also easier to clean?
Okay, so you have the medical mask, so the surgical mask and the N95 FFP23 mask. That kind of polypropylene melt blown material is the ideal gold standard, as you saw from the results that I showed in the slide. They are very effective at stopping virus from leaving as well as coming in towards your face. Then you have the cloth masks, you know, the bandanas made out of cotton or nylon. If you multi-layer those masks, like you see in the CDC video, and then you tuck the sides in, okay, then you'll get more protection from even just a normal cloth cotton mask as well. Now, there has been some talk about this by the CDC to actually wear two masks. So in fact, you wear like an N95 mask here, and then you actually wear your surgical mask over the top of that, like this, okay? And the idea behind that is that the surgical mask is more disposable than this N95 mask. You protect this more with a cheaper disposable surgical mask. And you might even consider using a cloth mask on the front and keeping this more protective mask more protected so you can keep using it again. But again, if you hang this in the sun for 8 hours, okay, the RNA of the virus will fall apart due to the UV light exposure from the sun as well. But make sure you label the mask. You don't want to mix up the mask between different healthcare workers.
Yeah, great, thanks. I guess healthcare workers can take masks with them home and then hang them at home separately.
Yeah, they hang them at home and they can take them home. Yeah, so I mean, this is a precious resource. Don't throw them away. If you can disinfect them, disinfect them. And you know, it's not 100%, but it's better than nothing.
Great, thank you very much. There are other questions about ventilation and in terms of transmission to children. I think they're quite big questions to tackle really at this stage.
Sure.
But one of the questions which I don't understand myself, but I'm sure you will, what is the benefit of heterogeneous prime Boston vaccine? I don't know that,
Okay, so what they're talking about is the mix and match vaccine approach, which they're trialing in China and also the UK at the moment. So like I said, if you have the AstraZeneca vaccine that induces a better antibody response, as it induces a better T-cell response, you might want to have one Pfizer and one AstraZeneca. Okay, so they boost different arms of the immune system. Also, if you have a vaccine that's designed to the Wuhan virus, but you have a booster, say a Pfizer booster designed to the South African virus, then your first dose AstraZeneca Wuhan vaccine can be actually boosted by the Pfizer vaccine that also covers the South African variant as well. So you increase your coverage of different variants. If, say, for example, AstraZeneca hasn't produced a South African specific booster, but Pfizer has. So you can broaden the spectrum of protection with a heterologous prime boost vaccine approach. The other thing is that some vaccines may not be very immunogenic in some people. So some people may not react to the AstraZeneca vaccine. There's several healthcare workers who have actually been tested post-vaccine, post 2 doses of vaccine, the Pfizer vaccine, and not responded with any antibodies. So they had the Pfizer in January, Pfizer in February, got tested in March for antibodies, no response. So you might then use an AstraZeneca vaccine as a booster, 3rd dose, and try to induce an antibody response in those who haven't responded to the Pfizer vaccine, for example, or vice versa. If they don't respond to AstraZeneca, they might respond to Pfizer. So there's several advantages of mixing and matching vaccines. And in fact, we do that already for some vaccines. For example, you know, my son had the pneumococcal vaccine, the Prevnar 7, Synflorix 10, Prevnar 13 when we were in Singapore, because the vaccine kept changing every few months. And he was fine. And so you expand the spectrum of coverage by Prevnar 13, but the other vaccines have primed the vaccine response already for the first 7 or 10 antigens that are also included in the 13 antigen Prevnar 13. So the prime boost vaccine response can improve your coverage, can also improve your immunogenicity. If you don't respond to the first vaccine, you might respond to the second one.
What would be the ideal time frame between the two vaccines?
So we're not sure yet. So AstraZeneca does have some data showing, you know, second dose response is good up after so many days. The other vaccine trials haven't shown that because they've stuck really strictly to their vaccine schedule, you know, 3 weeks or 4 weeks. And that really depends on individuals as well, because your first dose immune response will then set up the response to the booster second dose later on. Now, some people might respond after like 20 days, 10 days, or 30 days. And then that longer dose interval for the second one is more beneficial if you've actually optimized your first dose response. Okay, so if they're too close together, that's why most of the trials are 3 weeks to 4 weeks. If they're too close together, they don't actually augment your first initial response. It's almost like, you know, a double dose first dose, but you don't have that second dose booster if that interval is not long enough to generate that mature immune response later on.
Great, thanks. Just a question about Novavax. Do you think that is the most effective against most variants?
I think the Pfizer is the one that's been tested against most variants, and it seems to be the most effective against those variants. The Novavax was tested in South Africa in HIV positive and HIV negative patients, and it shows about 50% with HIV positive patients and about 60% with HIV negative patients. Against the other variants, I haven't seen a lot of data, but I suspect that all these vaccines will produce some immune response that's protective against severe disease and death. So I think if you take any of them, as long as you respond to at least one dose, the first dose at least, then you should be protected against more severe death from COVID-19, any variant.
Great, thank you. And in terms of the updates to the AstraZeneca vaccine, which would be the most, actually probably the only one that would be actually sent to Yemen, how often do they get updated?
So, I mean, like the flu vaccine, these vaccines, the adenovirus vaccines, the mRNA vaccines are actually easier to update, but then you have to manufacture it at large scale. So the estimates we've got from some of these manufacturers like AstraZeneca and Pfizer, you can actually update the vaccine within a few weeks. But then you have to manufacture it and then ship it to all these different countries, which may take 3 to 6 months. And in that time, the virus may have changed again.
Right. True.
So although the planned update is for the South African variant in autumn, AstraZeneca and Pfizer originally said they're going to do, but now they've pulled back on that. If this Indian variant becomes more predominant, then we have to target the Indian variant more than the South African variant, because in fact, the one that's surging has different mutations compared to the South African variant, unless they can combine all of them together. So, you know, have the 484K, the 484Q, L452R, 501Y, all in one vaccine. Then, of course, another variant could arise as well later on, even during manufacture.
Great, thank you very much.
Sorry, did you get that? Because you froze a bit. So did you catch that? Or
yeah, I did, and I hope the audience did catch that. Yeah, thank you very much. One last question. Actually, it's probably a theory by one of the person asking. This person asking, there was at the beginning of the all these events, there was a strange respiratory and fever a disease in the south of Yemen, which nobody knew what it was, whether it was COVID or something else, or nobody knows. So this person is asking, is there a chance that could have been a link? Bear in mind, this was really early on. That's even before the Wuhan, or maybe at the beginning of the Wuhan breakout.
So when was that actually? Was that last, like November 2019? Maybe?
Yes, October, November, yeah.
So what you need to do is, if you have that sample in a freezer somewhere, hopefully it's in a freezer, you can send it through, you know, WHO, Gavi, or one of the other collaborating centers, and get them to do full genome sequencing on it. And then you can actually then blast it and see what that pathogen was. And if it's a novel pathogen, you have to do some further work on it. And so if you go through Gavi or one of the WHO contacts to try and get that back to the UK, the COG UK team could do it for you. You could answer that question, because obviously now we're really hyper vigilant about any new pathogen that might be emerging from anywhere. So if you're seeing an outbreak of pneumonia, and try and get samples frozen whilst you contact people in WHO to get that sample to a sequencing lab somewhere in the world. The closest one for Yemen might be one of the Middle East and South African countries, that have this capability of sequencing.
That's great, because it just highlights the importance of sampling in these countries where the facilities are poor and there is widespread and there is very close contact of personnel and poor hygiene. So it's almost like a sitting duck for pandemics.
I mean, also the interaction with the animal environment as well. So if you have lots of contact with animals, you might get a animal to human transmission, like this bat to human for COVID, bird to human for avian flu, swine to human for the pandemic flu back in 2009. You don't know where the next one's going to come. So in fact, a lot of these rural environments are actually mixing vessels for human and animal infections that can cross between hosts and then cause the next pandemic.
Thank you very much. And we'll just finish with this one. Let's say, for example, somebody wants to sample, take a sample from an unknown respiratory disease in Yemen. What would be the best way to sample it, store it, and transport it?
Okay, so if you are faced with this situation, you want to make sure you wear your PPE because you don't know what you're dealing with. Okay, so, you know, mask or double mask, eyes, visor, and then gown and gloves to stop droplet transmission, splashing. Take a swab, but make sure you have a virus transport medium tube, not the solid gel tubes you have for microbiology. Okay, there's blue top, black top tubes. You don't want those. You want the fluid medium. So it's a straw colored fluid, pink fluid. Take the swab and then back of the throat, ideally, or through the nose to the back of the throat. That's quite uncomfortable. But back of the throat is easier. Swirl it around, rotate it, and then straight into that virus transport medium. Now the virus most viruses will survive some time at 4 degrees inside that fluid. But to really preserve it for a long time, you want to put it into the -80. Now I think most of the labs in Yemen may not have a -80, but even a -20, you know, like a home freezer, -20 is better than a fridge. And then it might stay stable if it's RNA for maybe, you know, a few months at -20. It'll stay more stable for 6 to 12 months at -80. But it may stay stable for like a few weeks at 4 degrees in the fridge. And in that time, you want to contact a courier or a lab to hopefully pay for a courier for you and get it transported on dry ice. They can send the dry ice to you with the courier, which is, you know, dry ice is about -80, and send that to the lab that will do the sequencing for you.
That's great. Thank you very much. We call on our colleagues on Aden, which is the southern city, which is the hub, which is a port and have a functioning airport, and has seen quite a few breakout of diseases. To take note of these recommendations and you know, try and put samples out there so that we can know what's going on in Yemen. We would be happy to link people to various agencies if they get in touch.
So I think the team in Aden do have the MSF equipment, do they not? Yeah, so true. I think they gave you some freezers as well, maybe -20, not -80, but that would be a start.
Absolutely. Thank you very much, Stan, for this amazing talk. As you can see by the questions and the volume, there's a lot of interest. Even though we ran out of time, we could talk forever because this sort of information is what is lacking on the ground. Expert input is really valuable. We really thank you for your time to be with us today.
Yeah, no, I'm very happy to share my email address with the Yemenis colleagues if they want any further questions answering, because there are lots of questions there which we can't cover at the moment. And I'll see what I can do.
That's very kind of you. Thank you very much. We will. You know, would you be happy if we share some of your slides?
Yes, I think you can copy. I think you can share all my slides. But let me just have a quick look through them so I don't get, you know, breach any copyrights. But I'll send you a sanitized version through Mubasher to INTERYem.
Oh, great! Thank you very much. Especially the slides which summarize the treatments, summarize the.
Yeah, those are from our hospital. You can share those. I can share those independently with you.
Absolutely great. Thank you very much. Thank you very much for starting. Thank you very much for your time. Thank you. Thank you very much, everyone, for bearing with us, and thank you for your interaction and your valuable questions and making this session very useful for our friends and colleagues in Yemen. And also, I'd like to thank the next speaker, Dr. INTERYem Babraik, for her patience and also allowing us to delay her presentation by half an hour. Thank you very much for that. Dr. Babraik will be talking to us about COVID surveillance in Yemen. It's a very specific topic to Yemen. Dr. INTERYem Babraik has worked in Yemen for many years. She has very, very hands-on experience with the health system in Yemen. She's currently a lecturer in global health at the University in Leeds. At the same time, she does a lot of COVID education and COVID-related talks for Yemen. I'd like to welcome Dr. INTERYem Babraik, and I would like to say thank you very much for coming again, because we have had you previously on some other INTERYem conferences.
Thank you very much. Thank you. Thanks for INTERYem for having this important conference. Very interesting presentation. And before I start, my presentation today will be talking about the surveillance in humanitarian setting, particularly the context of Yemen. But before I start, I want just to make few points related to the previous presentation. Yes, there is Yemeni specimen has been sent from Yemeni patient through the WHO to identify what type of variants we have in Yemen. We haven't got any data about the result, but the WHO has done this already. We are hoping not to have new variants. The other thing is the point that we don't have a virologist in Yemen. Actually, Yemen had some very efficient virologist who established virology department in University of Sana'a and University of Aden. But because of the instability in the country, many of them have left and they are now working in the Gulf region. Unfortunately, that's a consequence of the war situation. The other point which I wanted to mention is sometimes, yes, the speakers, they talk about things which might not be easy for Yemen to be implemented. So for example, when we talk about the opportunity to have different vaccines, unfortunately, Yemen is relying on what the COVAX is sending to them. So we don't have, not everything we can hear in the conference, we can implement it, but it's really good for the physicians and the doctors and the health workers to be aware about the pros and cons of things. But as the previous Professor Julian mentioned, having this vaccine, the one which is available in Yemen, AstraZeneca, is protecting against the fatality. Maybe we are not in a position to completely prevent the transmission of the infection, but having something is much better until we know what is, to what extent the AstraZeneca would be effective against the variants which is currently in Yemen. So moving from that interesting presentation to my presentation, I'll be talking about the diagnosis of COVID-19 at the community service, at the community level in Yemen. And that's what we call it, the surveillance. I'm going to give a little bit of background, what is the surveillance, before I explain what is the situation of the current diagnosis of COVID-19 in Yemen. But before I start, I want just to give a little bit of context. You know that Yemen is an unstable condition. It has many people who have been displaced internally from the places where the war or the bombing or the unstable condition have affected their villages, and they move to other places. And this is a picture of one of the camp that you can see people are there is no infrastructure, just sitting in tent. So there is no way for them to have social distancing. If COVID-19 come to a situation like this camp, you can easily find many of those people affected. Face masking is going to be very hard to be applied here when people, or even washing hands, when people don't have access to water. And this is just one of the families has been affected by the internal displacement. They don't even have a tent to live in. So the children are sleeping in the floor. This is just one picture of the rubbish is being collected in different places in Yemen. And all those, just to remind us that we are talking about a different situation, very difficult and unstable condition. So in Yemen, what happened is, we are now in the second wave based on the figures that we have, and I'm going to explain whether we do, how confident we feel about the data, whether this is a second wave or this is a third wave, and why. So we found here the intensive care unit is fully occupied in all the health facilities in different parts of Yemen, to the extent that you can find COVID patients are sleeping on the floor. And this is a picture have been taken from one of the isolation center in Taiz governorate. So there is no space for them in any place in the hospital. So that's where they sit until they find a space. And that's led into group of people who die and their bodies poured in the same day, and this is a picture being taken from the same governorate. This is what we have documented so far. So this is just to remind us what context we are talking about. So what is surveillance and what is the situation of surveillance in Yemen and why this is important? Just to remind you what we call it the epidemiological triangle. In order to control any epidemic, we want to have some information about three important aspects of or causes of this epidemic. One is the agent, and that's where the talk by Julian, Professor Julian comes. We want to understand what variant it is, what virus it is there, but also we want to know the nature of the host. Can anyone get it? If I get the vaccine, what is my chance to get this virus again? What is the immunity situation of this person? Does pregnant women protected? Does children, do children are protected? Some of the questions came in the chat about that. So all those are new information for us about COVID, but also what environment would make that virus cause the epidemic more or less than others? And there is an interaction between these three. In order to get this information, we actually need some system to collect information. And that system, we call it the surveillance system. The surveillance system is a system which collects information about who is infected, what age, what gender, where they live, what's their situation, do they have diabetes, do they don't have diabetes, other comorbidity and so on. But they also get some specimen from those hosts to identify what virus is causing this, what variant is causing that, and what environment. Is it hot countries are having less prevalence of COVID-19 than the UK and the cold countries and so on. So this is actually what the surveillance system is trying to look at. So this is to highlight, without knowing this triangle or this three dimension, it will be very difficult to understand what is going on, and it's very difficult to propose an action. So the role of the surveillance system actually and the testing, I consider it, and many epidemiologists and people who work in public health, they consider it as, you know, when you throw a net under the sea and you want to get this net collect as many fish as possible under the sea. You don't know what's going on under this boat. So, but the bigger your net is, the stronger your net is, you will be able to get as many things under the water. And then you might get something like different type of fish. You might get baby fish. You might get different type of sea animals. You might get rubbish. You might get so many things. It doesn't matter. So the screening system or the surveillance system is actually trying to get as many cases as possible. It's okay if we can get some people who are not COVID patient, but they have similar symptoms. It's better to get them, protect the community from transmitting the infection from those people to others. It's much better than getting, you know, the small net that might not give you the full picture. And why we need such surveillance system? Because this is something important to understand in many diseases, and particularly in COVID-19, that you only get those who are coming to the hospitals are those who are either having severe symptoms or they are close to die, or maybe they die, so we register them as a death because of COVID-19. But these two represent only the top of the iceberg. So you can see this is a small part, but if there is a ship going on this direction, what you can't see in the epidemic control is this big part under the sea. And this big part under the sea is actually the mild symptoms, people who have mild symptoms, or they are asymptomatic, so they've got COVID, they don't come to you. So the more you are able to get things under the water surface, the better you will be able to protect them and predict them and prevent them from transmitting the infection to the others. So this is just to conclude why we need a good surveillance system. So if a patient comes to me with a with the severe cases being admitted to the intensive care unit or have any symptoms related to COVID, is actually, this is the top of the iceberg. But I have to go and trace all the contacts because I'm looking for the part under the sea, the things that I cannot see, which is the mild and the moderate symptoms. And that's actually what the surveillance system is doing, not only registering the mortality and the severe cases, because they do come to the health facilities anyway. We would like to get them and trace the contacts in order to widen our net in order to get as many cases and prevent the transmission of the infection. So based on that information, you can imagine if a country does not have good surveillance system, it means you are getting small, tiny part of the cases while you leave the majority of the cases to transmit the infection from one family to another family, from one village to another village, from a town to a town, until you have a real disaster in the community. So this is just to give a little bit background about why this is important. So in order to do the surveillance, if I would like to get that big, strong net, where I get the information from and what type of information I would need, we would like to get all the cases reported to the health facilities. Not only the isolation units, in many countries, low middle income countries, the cases are only reported from isolation units. We need to involve all the hospitals, even the private hospitals. All of them, they need to be linked with the data system that report there is a case, whether it is suspected cases or it is a confirmed case. We also need to get information from the community, and getting the information from the health facilities, we call it passive surveillance. You sit in your office and you get all the information about the cases coming from the health facilities, but that's not enough. You need also to go to the community and contact tracing, see who is got some symptoms in the community, but they haven't came to you. You can use the what we call a rapid response team, which I'm going to explain in the next few slides. You can also use the community leaders. You can use the school staff. You can use people who are running the mosque, any religious activity. Those people are important source of information for the surveillance system. Not because we want to attack people, but we want to get as many cases reported to stop them from transmitting the infection to their neighbors, to their family, and so on. But we also need to keep our eye on a particular people who are more likely to get the infection. So for example, the healthcare workers, you know for sure the healthcare workers will get the infection, whether it's mild or moderate and severe. So all the healthcare workers needs to be observed for all the symptoms, and the should be also diagnosed. If we don't do that, we are getting the infection to be transmitted from the healthcare workers to their patient, to their relatives. And then you can see everybody who is coming to the health facilities, if we don't monitor the healthcare workers, regularly checking their serology for, themselves for COVID-19, hospitals and health facilities can be source of infection to the whole community. And that's maybe people who are in Yemen, they would know why many cases being transmitted through the health facilities, because there is no proper system for screening the healthcare workers or even diagnosing them. I'm also aware that many healthcare workers, when they get infected with COVID-19, they don't even report it to their health facilities. So I only take sick leave without explaining what's going on, and they keep quiet, treat themselves in the house until they think they are okay, and then they go back to the health facilities without 100% being sure that they are free from COVID. There is also important source of information, which is the point of entry. People who are coming through the airport, the ships, and all those need to be monitored. At the moment, this is not taking place in many low middle income countries. And if we don't do that, any variants around the world can come to the country. And then we keep struggling with the cases. Also, any sample that come to the lab, even if it is a private lab, should be monitored also for what was the case presented with. In addition to monitoring the deaths, anybody who passed away during the COVID pandemic should be investigated, and there are some investigations that we use to identify whether it was suspected cases. But I would say the simplest investigation is to ask, what was the symptoms? What were the symptoms before the death? Is there anything to do with fever or the case definition? If yes, I would consider it as a case, and then do the same for the contact, all the contacts. So this is the theory about who should give the information. And during my discussion, I said the source of information could be the community, could be the clinics, could be the hospitals, could be COVID isolation units and centers, private health facilities, anybody sometimes calling to ask about any symptoms could be source of information. So there is a lot of source of information that we could get about in order to get proper surveillance system. So this is the theory. And I don't want to speak too much theory. I want to explain what is the situation of Yemen at the moment. But it's really important to get an idea about what should be in place. Then people can imagine how far we are from the perfect or the ideal. I wouldn't say the ideal. I would say the minimum for safety in Yemen. And before I go to the situation of Yemen, usually, in case of any epidemic, there are two teams working into the surveillance. So who is going to get the information from the community, from the hospitals, from all those sorts of data? Usually you have the surveillance team, and the surveillance team usually work with a specific capacity. So they have limited number of staff. However, in case of epidemic, not only in COVID, any epidemic, we usually need more people to get information. And that's what we call it the rapid response team. The rapid response team are extra people who are before the outbreak. Usually we recruit them. We, as part of the planning for preventing the epidemic or quick response to the epidemic, we recruit them and we train them and we keep them on hold. In case of any epidemic, we retrain them and then activate them and ask them to work in the case of epidemic, in case of epidemic, to go to the community, report cases, go to the hospitals, go to the health centers, speak to the social leaders in order to get as many cases, suspected cases, to be confirmed, take the sample and confirm the cases, but also trace the contact in order to control the epidemic. After the epidemic is being controlled, they go back to be in our list until a new epidemic happens. So countries that did manage to control the epidemic in the first, to get a proper surveillance system in the first wave and the second wave are those who have quick response from the rapid response team. Unfortunately, many countries did not use the rapid response team because they just realized they haven't had any epidemic for years, including high-income countries. So they haven't trained enough people, and that's why it takes them long time in order to get the surveillance system, the reporting of the cases and the contact done very well in the first wave. But they learned a lot for the second wave. So you can see some countries, the mortality in the second wave was, or the reporting in the second wave was much better than the first wave. But many countries haven't learned a lesson until now. And many countries, they are in the third wave and the rapid response team is not is not working properly. So what is the situation in Yemen? I'm going to explain that, but you keep in your mind that how important the rapid response team. They are extra people who would do the work in case of epidemic when you have thousands of cases, hundreds of cases are reported. So of course the surveillance team wouldn't be able to get the information from all those resources that we have mentioned during the epidemic. Now the WHO have 6 steps to help countries suppress the COVID-19, expanding the training for the health, public health force. So usually many people in many countries, you might be working in some specialties. In case of the epidemic, in case of the COVID-19, many people in many countries, they've been asked to leave their place and work in surveillance system. And this is recommended by the WHO because they will have the basic knowledge. It's much easier to train them than getting rapid response team from the community or people who have basic information about health. The plan was we would like to have a system that find every suspected case. If we can get every suspected case, this is how quickly we can suppress the COVID-19. And that's the basic job for the surveillance system. The third important step that to suppress the virus or the epidemic is testing capacity and the availability. We need really to improve the testing capacities. What's the situation in Yemen? It's really unfortunate to say there is only 10 labs are available in Yemen that cover 30 million population. So we are so far from getting the suppression of the COVID-19 because basic things are not there. And I'm going to explain why this is not there so far, because of training. People who are trained to do these services are not available, but also the money. The country is no, it wasn't a priority for the country to diagnose virology. And unfortunately, because we have a lot of bacterial infection, parasitic infection, so those were the main causes of mortality and morbidity. It is the first time that we have this epidemic caused by virus. So most of the labs, they are not ready. They were not, and they still are not ready to diagnose virology, diseases caused by viruses. So we don't have the basics in place. So when we talk about testing capacity, we are actually talking about establishing from scratch things like this. And that's why they are very expensive. And that's why the WHO couldn't have more than the 10 lab around the country. Also, the fourth step is to identify and adapt key facilities that to treat and isolate patients. And I think Yemen did very well. There was some equipment of the isolation units, but that's to do with the treatment of those, if you remember my triangle, to deal with the severe cases and the people in advanced stage. But what about the mild and the moderate cases? This was not in place because we couldn't trace the cases. The fifth step, which is really very important, is to quarantine contacts. If I am 100% sure this is a case, whether I do the test or I don't do the test, if you don't quarantine the contacts, those contacts will transmit the infection through the community. And who is the job of this, who can do this step is actually the surveillance team. But if the surveillance team is only small number of people, you would need rapid response team. If the rapid response team is not large number of people who could cover all those contacts, then this is not going, the epidemic is not going to be suppressed, is not going to be under control. The sixth important thing is refocus of the whole government to contain the COVID-19. Some countries, they did very well. When you can see the newsletter, everybody in the country is talking about COVID. All the education, all the programs, all the discussion, all the social activities talks about COVID-19 in order to control it. But in many countries, no, things are still taking place normally. You can see, we have seen conferences talking about cardiovascular problems, and as if there is no problem called COVID-19. And I'm going to explain the challenges that the country is facing. But one important thing is lack of awareness, because we didn't see by our eyes lots of people dying because of COVID. We did not see large number of patients being admitted to the hospital. Then people think, oh, no, Yemen is away from this epidemic. But when the cases are reported, when the deaths are increasing, then we go to identify there is a problem. But also for political reasons, many people know that it wasn't in the priority, and still it's not in the priority of the people who are politically leading part of the country to do that. But there was some trial. But if we would like to talk about suppression of COVID-19, we need the whole attention of the government. But we can't leave this point without talking about, well, if you have a country that people are dying because of hunger, because of malaria, because of other problems, you cannot stop everything and say, well, we want only to focus about COVID-19. Maybe that's another reason for why COVID-19 was not or is not having the full attention of the Yemeni government. Sorry. So in order to get the information for Yemen, this was the strategy published by the WHO in July 2020. They wanted to say, well, let's first of all monitor suspect and probable cases through the health facilities. We also would like to have a community surveillance through rapid response team. And we also want to have active case findings. We are not going to stay in the hospitals and health clinics waiting for people to report the cases, because as I said, there is a lot of reasons that people wouldn't report the cases to the health facilities. So we need to go and do active surveillance, active case findings in the community. So these are the three important recommended for Yemen in the first wave when last year, July 2020. They also said when the money would be available. For these three components, they are essential, or they are considered as essential steps. Step one, but later on there was a plan to have improving the lab testing data for Yemen when they have more capacity to equip and train people on more diagnosis and improve the diagnosis of cases. And later on, there was a plan to have seroprevalence studies. And the seroprevalence study, the aim is to identify how many people they had a response to COVID-19, and you could consider them as COVID positive. And this was done by the WHO as a study. They took Aden as an example of the cities, of the Yemeni cities, and they found that 27% of the population had COVID-19, even though the cases, as I'm going to explain in the next few slides, did not exceed until now 6,000 cases in total out of the 30,000 population. And that's emphasized that the fact we were talking about is the majority of the cases were asymptomatic or have mild symptoms, or maybe they had severe symptoms, but they did not report the cases to the health facilities, or they died without diagnosis of the cases. We only know little about the cases that have been diagnosed and confirmed. And let me go back to the triangle so you remember what am I talking about. So this is what I was referring to. The majority of the cases here were either mild or asymptomatic, or they passed away without being recognized as COVID-19, and we know little about those who had severe cases and being reported to the health facilities. Remember, the severe cases, some of them, they been treated at home, and maybe they go in the last moment to the hospital where there is no chance to diagnose them. So this is the situation in Yemen. It doesn't mean that COVID-19 is not available in Yemen, but we would like to know more. I'm going to skip these details. So in the second wave, now the WHO also announced they have 10 pillars in order to control COVID-19 in Yemen. And I'm not going to explain the 10 pillars in details, but one important thing is to have a coordination between the different parties and the different facilities in Yemen and the south and the north in order to have joint planning on response otherwise, but also better management of the budgeting, because otherwise this is not going, the suppression of the virus is not going to happen. The second thing is the risk communication and community engagement. And this is to talk to people in different places about the importance of raising public awareness, importance of having reporting the cases, because if people don't report to you, how many people, imagine how many people you would recruit them in order to go to the community. And sometimes when you go and say, well, you do have COVID-19 symptoms, and people deny that, no, I don't have, how are you gonna convince them to do the testing? So risk communication and community engagement is really important for us to understand what is the situation and then plan for it.
Dr. Miriam, thank you very much for this. Is this your last slide? Just to gauge how much questions we have for this.
Did I pass the time?
Yes, you have by 6 minutes.
Sorry.
Professor Ramirez need to go to another meeting after his slot.
No, actually, it's not my slide. It's not my last slide. I thought I still have 10 minutes.
Okay. So what I would say, if you want to finish the most important slide,
I can manage to do it, the key concept in 10 minutes.
Yes, please, that would be great.
Yes.
10 minutes,
yes.
Is that going to be, I think 10 minutes will be 25 minutes over your allotted time, or 20 minutes. Is that going to be okay with the next speaker? That's all I'm worried about. It's all interesting stuff.
Sorry, it's just because the situation is really complex, and I don't want the audience to, you tell them about the situation.
Okay, how about we go back to Professor Ramirez, and then we come back to your talk? Would that be okay, do you think? Or
I think the idea is going to be cut it. I think I can manage in 10 minutes to wrap what the situation is.
Okay, fine, carry on. Fine.
So the idea is, if you go back to the WHO website, you can see up to date, there is only 6,000 cases being reported as COVID, and the fatality rate is 19%. And that's really made us to think about, is that real numbers if we know all those background in place without having surveillance system, good surveillance system? I wouldn't trust these findings in general. So what we did is we implemented a study in 2 phases. The first phase was an exploratory study to go and see what is the testing capacity in the country, how the team is working. And one important thing we found is all those cases that are coming, they are actually coming from mainly from 3 governorates, Hadramaut, Taiz, and Aden. There is no data coming from the rest of the country. And further exploration for this, we identify that you can say 70% of the population in Yemen, we have no idea whether COVID is being in place or not. So that made us to explore, go further to the team, the surveillance system in Hadramaut, and ask them about how they report the cases, how they work in the system. But also we went to the WHO and the key stakeholders that are working in Yemen to see what is in place. Surprisingly, we found that the surveillance system, which was in the strategy of the WHO, they needed 500,000 US dollar to do it, but they have 100% gap in the fund. There is no fund for the surveillance system. So I'm not surprised to see why the reporting about Yemen is not in place. In order to do that surveillance system, they need national laboratories, and they need 15 million US dollar in order to cover that. But the deficit is 93%. So there was no hope for Yemen to get the proper testing. So what we also did, we did a workshop with people who are working in Hadramaut, and we asked them about the challenges, how they work, what are the opportunity, and what's the recommendation. So I'm going to summarize to you what they do is actually, the team in Hadramaut, they follow the WHO guideline in terms of what is the criteria, the follow-up. They have recruited rapid response team. However, they have only 5 members of the surveillance system team. And during the COVID-19, they had extra 16 people. So you can imagine there are 2 members of the rapid response team covering 275,000 population. So simply, this is not possible. So the team also, because of the limited number of staff, they rely on passive surveillance. There is no active surveillance being effective in the country. So the main challenges we did identify for COVID-19 surveillance in Yemen is actually weak registration, low quality of the data, delay of the notification, poor case findings, very, very poor contact tracing, and there is an issue of stigma. People don't report cases, and if they report cases, they report it to people and they ask them, please don't report it to anybody. No health insurance. For the rapid response team themselves, they did not have protective measures. So many of them, they get infected during doing the job. So that was a big challenge for them. Poor community awareness and adherence. People deny COVID-19, and if they get it, they just go without protecting themselves. They feel they are okay. They don't understand what is asymptomatic cases. The labs were overwhelmed, have limited testing capacity. There is also other epidemic, as some people mention, an outbreak. People get confused. Is that COVID? Is it malaria? Is it dengue fever? And so on. But also the team themselves, they did not have proper transportation. The insecurity because of instability, because of the war, because of bombing. There was also no national lockdown, which had to lead into more and more cases. So simply, these are the challenges of COVID-19 surveillance in Yemen. The second phase of this study is taking place now. We are exploring other governorates to understand more and more. We are hoping by the end of 2021 to get this data published in a peer-reviewed journal to enable the country and highlight the importance and the need of this country to get ready for any viral infection. As the previous speaker has mentioned, this might not going to be the end of COVID-19. So we are hoping by publishing this data, we raise the awareness of the global health to the importance of addressing those challenges. We have a clear recommendation, but the study is not ready yet. We are hoping to do it by the end of September. And that's all from my side. Thank you very much for listening. And I wish I did manage to do it in 10 minutes.
Dr. Miriam, thank you very much. You actually did it in less than 10 minutes. That's really impressive. You know, your talk is amazing. It touches on really, really good stuff that is very relevant to people on the ground in Yemen. And it's actually generated a lot of relevant questions. What we'll do, if you're okay, we'll just maybe run through some questions and then we come back to some later, if that's okay with you.
Yes, sure.
One of the questions is a little bit strange, but some people in Yemen think that everybody in Yemen has actually been infected with COVID and have reached herd immunity. So if that's the case, then there's no point in us doing all of this. There's no need for any surveillance and there's no need for vaccine, but we still see people dying on the ground. So it's quite a strange question, but I hope you may have a brief answer for people who might think this way.
Yes, I think that's really related to that's why I know these beliefs it's there in Yemen, and that's the reason why I started with explaining the situation of Yemen and how COVID is in place. Actually, you can't say everybody in Yemen has got COVID. The serology test from the serological study from the WHO says the maximum we could have been infected in Yemen is 27%. And even if we have the confidence interval of 5%, I would say the maximum would be 35% of people. Without having the surveillance system, you can't really do that. However, I'm aware that the WHO is planning to do this serology test in other cities as well, so we can get a better picture of what is the situation there. So I would say I don't think Yemen has the herd immunity. Even if you get infected once, the immunity does not last for long. So you still need the vaccine to protect yourself from the COVID-19 consequences.
Thank you very much. And one question somebody is asking about testing, because really surveillance require accurate testing. How do they deal with the, running out of, for example, reagents or, facilities and stuff? Is there someone they can contact? Is this process centralized? People in different cities in Yemen can ask someone, and for example, top up of supplies and so on.
Yeah, it's actually the situation was really, really very difficult. There is only 10 labs are prepared to test for COVID-19 across the country, and they are not equally distributed across the whole country. There are situations in the first wave where people are running out of reagent, maybe the lab people who were trained. You can imagine in one of the lab there were only 2 people trained to deal with the test. One of them get infected, and the second one was overwhelmed. So the country was not ready to deal with the virology. Now the plan is much better. The plan, not the implementation of the plan from the WHO plan, but because of the budget, the WHO is not able to raise the capacity of Yemen to do the testing. We are just waiting for donation, and that's why during the conferences, we want to transmit the message. This is the situation in Yemen. We are keen to get this communicated to others, hoping that we're gonna get some more resources. But I also wanted people to understand, without a proper surveillance system, you cannot just call for intensive care unit. You cannot just call donation for oxygen. You want to prevent more and more people from being in a situation to need the oxygen. So those who are making decision need to be aware of why it is important. Testing is important, if you see what I mean. So there is a need to raise awareness among people who are making decision to give the donation and those who are deciding where the donation should go.
Absolutely. Thank you very much for highlighting this. We hope our colleagues in Yemen will find this useful, and we'll be happy for them to get in touch with us, and we can link them with the appropriate agencies. If you're okay, what we'll do, any more questions, we will have a chat with them later. But to be honest with you, the last answer has actually touched on 2 or 3 questions together. But if the people asking the questions feel that they still want to ask them again or rephrase them in a different way, please do so, and we will promise you will come back to them after the next talk. Thank you very much, Dr. INTERYem. Thank you for your time. Please allow me to go to our next speaker. Our guest is from all the way from Mexico, Professor Jose Ramirez. He will be talking to us about the new antibiotics, challenges and opportunities. So a little bit different from COVID. However, it goes nicely with our theme, which are the challenges of infection in Yemen. Professor Jose Ramirez joined the Autonomous University of New Lyon with the position of full professor at the Faculty of Chemical Sciences. He is the founder of the Nanobiotechnology research group, where he is the main researcher. As of October 2015, he was appointed as the director of this new center. One of the most important research areas in his group is the development of novel medical therapies to combat infectious diseases. Let me please welcome Professor Ramirez. And in advance, I would like to thank you very much for being patient with us. I know you have meetings to go to after this, but we're really, really thankful for you being with us today. And I would like to give you the mic. Thank you.
Are incredible. I'm going to be a little bit of a disappointment that I'm not going to talk about COVID, but I will talk about some of our recent developments in the development of new therapeutics, especially for to fight infectious diseases and diseases caused by resistant bacteria. And so I'm gonna give you a very brief overview. So I'm from our center is, and I am also from Mexico. This is some of the views from our city. It's a mountainous city, so we are surrounded by mountains all over the place, and there's a lot of extreme sports that are done, climbing different terrains. This is a picture of the main library in our university, which was designed by one of the most important architects in the new era of Mexico, which is Dr. Legorreta. And this is our new research center on biotechnology and nanotechnology. It's a center that tries to involve interdisciplinary research between nanotechnology and biotechnology for many different applications, and that application aspect is involved in the engineering part of it. And so right now we are about 25 scientists that are linked to the center, and which our main goal is to do interdisciplinary research for different applications. Especially in my case, I do nanobiotechnology or nanobioengineering for the development of therapeutics. And I like to start my talk by just talking a little bit of the aspects of the importance of interdisciplinary research. As we know, application comes from engineering, and usually when you think about engineering, you think about some of the basic concepts of the basic science that you are trying to get a toolbox that you can then use. For example, the toolbox in mechanical engineering was the classical mechanics of Newton, and then you develop that toolbox that then you can use, and then you get mechanical engineering, and so on. No? So these toolboxes are being developed in the nano fields and the biology fields can then be exploited, and we have a very fertile landscape in the development of basic science in all of these research areas, so that we can then apply them and do nanobioengineering for different aspects. But when we think about the development of technology through engineering, we might think about telecommunications or going to space or development of new movement aspects. But we hardly ever talk about the technology in our drugs or in our pharmaceuticals, right? So I think that one of the key aspects for us to have pharmaceutics that are intelligent or that do not have all of these side effects and all of this, and for them to be much more efficient, we need to think about three things, right? First of all, and that's not a lot of engineering, that's not a lot more basic science, is the development of excellent active ingredients. So we need to develop, and that's part of mostly part of the chemists, pharmaceuticals, and biologists, so that they develop efficient active ingredients to treat a specific disease. But then we need to think about all of the delivery of the drug, right? Once you have an active ingredient, then what is the target and where is the target located in your body for that drug to go and perform its job, right? And then afterwards is all of the part of the clearing of the drug. You want the drug to go do its job, cure you from the disease or from the symptoms, and then the drug be out of your body immediately or in a short time afterwards. And so this is what I call intelligent drugs, right? So in my research group, we try to develop intelligent therapeutics that kind of cover these three aspects. And so I'm going to take you and we do it by working in the interface of engineering, biotech, and nanotech. And so I'm going to take you through a very brief path of where of how we have developed different therapeutics. In my research group, we focus on the development of therapeutics through this kind of like this perspective, but we also do a lot of sustainable processes, which is also has to do a lot with the development of scaling up biological process to produce therapeutics, which I'm not going to talk to you about in this talk. But so the development of therapeutics to treat infectious diseases, it's the history where how I got involved in it is the fact that when I was doing my PhD, I came into a group, a nanotechnology group, a group that developed basically nanoparticles, metallic nanoparticles, biomaterials. And one of the key aspect that my advisor wanted to tackle, especially from a special grant that we got, and my advisor was Nicolas Pepas and Dr. Yacoman from Mexico. And they said, there's been an increase in the resistance of bacteria. And this is also before COVID, this was one of the main problems that the WHO was very, very scared of, right? The fact that we developed antibiotics around the 40s, and we had this like golden age of the development of new therapies. But then we kind of stopped developing new therapeutics for around 40 to 50 years, which is around 1960 to 2000, 2010. And so we started, since the beginning, we started seeing this kind of graphs, right? Where there's been much more and more incidents in the infections that are treated in patients. And these infections, we see a lot of bacteria that are resistant to the different antibiotics that we have developed. So, and with the years, there's much more incidence of this bacteria. And so if we don't develop new drugs and we start seeing this type of graphs more and more, then we are going to, there's going to be a point, and we have reached, we are at the brink of it, where most of the antibiotics will not work against the infections that are common to the population, right? And so when I was working in my PhD, we got a grant from a Mexican company called Peñoles, which is one of the biggest companies that produce silver in the world. And I did my PhD at the University of Texas at Austin. And so I started working with silver because they wanted to see if we could find other applications of silver, which, as you might know, silver is a very mysterious component. It's part of the folklore and the culture of many civilizations, right? Such as the fact that you can only kill a vampire with a silver with a silver sword, right? But what about silver, which has a lot of it's anciently known for having antimicrobial properties, and it was one of the first antimicrobials for being developed, or at least reported by Hippocrates, where he said, you can use silver to treat wounds and to keep your food safer. And he was talking mostly about metallic silver. But what about silver in its nanoparticle form? So as you might know, or if you don't know, you can look at a little bit of some papers on silver nanoparticles, and you can see that silver nanoparticles are very easy to use, very easy to produce. All you need is metallic salt, which is silver nitrate. Then you do a reducing agent. In those times, a long time ago, we used sodium borohydride. But now you can use any tea, any leaves that you make a tea out of. It has antioxidant properties, so it's kind of like you can do a redox reaction, and then you can reduce your silver and produce your silver nitride and your silver ions and produce silver nanoparticles. And we kind of like did some experiments. Actually, I came to do a research stay in Mexico when I was doing my PhD, and we did a kind of like we proved silver through a range of bacteria. So you can see we have E. coli, Pseudomonas, typhus, cholera, and we will see that it will have a broad spectrum of effects at different concentrations. And then my group, since it was a nanotechnology group, it did a lot of electron microscopy. So we did a very thorough study on electron microscopy to see how the morphology of bacteria would change and why it would have such big effects and broad effects on different types of bacteria. And we mostly came up with a very rough mechanism, and mostly directed to the fact that nanoparticles would interact with different biological components, and they will attach to the membrane. And the membrane was one of the mostly disrupted parts of the cell. And so this had a lot of interest in the research community. And so when I did my postdoc in Jim Collins's lab in Boston, MIT, and University of Boston, I started to see what was the biology behind some of the metals, especially silver. And that was one of the initial projects that he proposed to me. He tried to work with systems biology, which basically systems biology to try to decipher these antimicrobial mechanisms. And we can define it as kind of like the impressionism, right? If you look at the arts by Monet and Manet, they do what they do is they do all these brushes, separate brushes that you can only see them when you go back a little bit behind, and you can see the whole composition. But if you go inside and look at one of the brushes, you can't really see what's going on in the picture. And that's what systems biology is, right? If you take a look at one gene, you might have a lot of high definition on how that gene works and what does this gene interact with other genes, but you don't really have a whole picture of what's going on when you tackle a cell with an antibiotic, or when you treat it with a specific chemical component. You don't see what's going on everywhere else. You might just see this high definition. It's two different perspectives, right? But systems biology, I think, gives you the ability to kind of have a broad picture, but gives you very interesting information. And so we started working with this systems biology techniques, all of the omics, metabolomics, transcriptomics, proteomics, etc. And so we tried to come up with a mechanism of bacterial action of silver in this case. And so we just very briefly, we tested with silver nanoparticles and with silver ions. We tested different, in this case, we did all of our work in model bacteria, which is E. coli. And so we first tested it with we found out the bactericidal concentration. Then this was actually afterwards for the paper, but we did a lot of toxicological work, and we found out that more or less the LD50 of mice was around between 240 and 120 micromolar, and we treated bacteria with around 20 to 30 micromolar of silver. So we were kind of like on spot. And we came up with, well, we did a lot of work, and we came up with a whole mechanism of how silver targets the cells, which tackles the membrane, which had already seen just in INTERYem, but it also tackles some other biochemical pathways, such as the Fenton reactions in E. coli, and also the TCA cycle and the production of hydroxyl radicals. But mostly, from an interesting engineering perspective, I started seeing the fact that a disruption in the outer membrane would be a really cool way to use silver to kind of potentiate antibiotics. If you have an increase in permeability of the cell or the integrity of the membrane, which is basically the barrier, then you can have maybe potentiation of antibiotics just by this mechanical disruption. And so we actually tried to use vancomycin in gram-negative bacteria and try to potentiate this increase in permeability in the cell that you would observe. As you might know, vancomycin it only attacks gram-positive bacteria because they have they don't have this double membrane that the gram-negative bacteria have, and which vancomycin is not effective against them. But using this perturbation of the membrane, we were able to get vancomycin inside the cell using silver, and then having vancomycin spread or widen its antimicrobial action now against gram-negative bacteria. And you can see we did this very cool experiment where we did it in mice. We infected them with a systemic infection with a pathogenic E. coli. And then we did various treatments. As you can see, most of the mice died with the vancomycin, with just the control. But then when we used the vancomycin and silver, we were able to save 90% of the mice. So that was a really cool experiment. And then we did some really nice experiments with resensitization of resistant strains. Could we use this increasing permeability to have resistant strains become less resistant, or at least get them back close to the sensitivity that wild type strains had? And so we did that. We had this wild type E. coli and this resistant mutant tetracycline, which you can see uses almost 3 times more, or 3.5 times more, the concentration of tetracycline needed to kill it. But then when we tested it with adding small amounts of silver, we were able to get the strain go back to its wild type sensitivity. And so this we had our 5 minutes of fame. We were in all the media. And so afterwards, when I came to Mexico to start my lab, I wanted to keep doing other strategies to develop novel antimicrobial agents, especially mixing nanotech and biotechnology and engineering. And so I kind of like broke my line of research, at least in the development of therapeutics, in 3 different aspects. One of them was creating these synergistic cocktails, not just with silver, but with other components, try to see if we could get combinatorial treatments to work better and to have much better antibiotics with what we have right now, without the need to develop new antibiotics. Then I did all the part. We have an aspect on engineering or delivery systems of these drugs, and try to see if we can come up with novel delivery system to make these antibiotics more intelligent. And then we have another line of research, which is trying to discover new antimicrobials in a very natural way, which I will speak to you briefly. But first, the synergistic cocktails, we try to use nanoparticles of silver produced by, now that we have all this tendency of green chemistry and environmental chemistry. So we did this antimicrobials, silver nanoparticles using bacterial extract. So what we did is we grew bacteria, and then that bacteria in itself has this redox components in the culture media. And so we were able to produce very nice nanoparticles of various sizes, and then treat with silver nanoparticles non-growing cells. So that was one of our key aspects, that we were able to kill bacteria which were non-growing. So that's one of the biggest challenges in many infections, which is sometimes the infections are located at sites where bacteria form biofilms, or have the ability to increase the population of non-growing cells or metabolically non-active cells. And so these are very hard to treat. No antibiotic now is able to kill this kind of bacteria in these metabolic states. But with silver, we are able to at least kill between 1 and 2 logs at a little bit higher concentrations of non-growing cells. And then we, with a PhD student of mine, we said, could we come up with other metals which are similar to silver, at least transition metals, which are less toxic, or at least less toxic to clinicians, right? Because they're always saying, how are you going to take silver? And I agree. I mean, it's not, it's a metal that we actually don't, our system is not able to process. But there are other metals, transition metals, that our system is able to process, such as micronutrients, which are called. Many of these metals are inclusive, necessary for many biological aspects in our body. And so even if in your cereal boxes, you can find a lot of what it says, this has vitamins and minerals. These are the minerals that these cereals and many of our foods have on, which are micronutrients. And so we use micronutrients to try to potentiate not just silver, but other antibiotics. For example, we came up with silver and micronutrients such as copper or nickel or zinc or cadmium, where cadmium was actually our positive control. And so we were able to see that we could obtain 100% inhibition and kind of like decrease the concentration of our silver. For example, we came out with 0.5 the MIC, 50% the MIC, and 50% the nickel, which give you 100% inhibition. But then even 0.25% and 50% of silver would give you also near to 100% inhibition, and that would be with all of the other strains. And we're out all the other concentrations, at least in this top right corner, and we can see that we will see a lot of synergy between this combination of metals. But we also did viability, and we will see that in a lot of in most of these combinations, there's no toxicity on cells. And then we did some synergy, not just with silver, but also with a lot of the micronutrients, with different antibiotics such as kanamycin and copper, cobalt, or nickel. And we can see that we see all of this percentage of inhibition at some concentrations of kanamycin and these micronutrients. So we will see that we'll have very cool synergistic effects. And we did this experiment with kanamycin and zinc on the right, and we will see that we will get a synergistic effect in curing a skin infection. We also explored the Mycobacterium tuberculosis field, and we were able to obtain, at least for resistant Mycobacterium tuberculosis, we were able to couple silver nitrite or the silver salts with some of the first line tuberculosis drugs, and we were able to see very interesting synergistic effects, and for us to allow us to do combinatorial therapies using silver and some of the first line therapeutics against resistant strains. And then I want to talk to you a little bit about some of the engineering that we have done on therapeutics. As my ex-advisor, Nicolas Pepas, would say, in most cases, the most complex part of developing therapeutics is to design its appropriate delivery system. And that's true even for COVID, which is, I think, the only time that I'm going to mention it. For COVID, there are many, including silver, probably kills or inhibits COVID. But the thing comes in, how are you going to deliver this thing so that many of these drugs are actually very toxic also for humans, right? So you're trying to avoid this problem, like in this cartoon, right? This guy goes to the doctor and says, listen, when the side effects of this medication kick in, you'll forget what was wrong in the first place. And sometimes this is what actually happens, even with COVID vaccines, right? Which I was vaccinated a couple of weeks ago. And the side effects were horrible from the Pfizer vaccine. And so there are current uses of antibiotics. Many antibiotics have a lot of different side effects, which some of them are serious. So the ideal for us is to have this efficient antibiotic, but also have an amazing delivery system that will have its GPS and its shield to protect it from autoimmune responses, etc. So we came up with these microbial exopolymers. So microbial exopolymers are really cool materials that are made from bacteria, and they're basically just polymers that are excreted by bacteria. And we found these exopolymers in a fungus, Rhodotula mucilaginosa, which is autochthonous from the northeast of Mexico, here in my state. We found this strain in a terrain that has high presence of metals, and this strain would produce a lot of exopolymers or exopolysaccharides in the presence of metals, of high and precious metals. And so I thought, this polymer must have some kind of compatibility with different metals, so that, and also is biocompatible, so that it might be a cool substrate to kind of grow maybe nanoparticles or grow antimicrobial therapeutics, so that we can use them for treatment. And that was exactly what we did. So these exopolysaccharides have these functional groups that can be used to grow metal seeds, and then which develop into nanoparticles. And so we did this really. We put our exopolymer or our polymer, and we did the synthesis of the nanoparticles inside the polymer, and you can see that the polymer is full of nanoparticles, of silver nanoparticles in this case. And then we would see that it would have really nice properties, antimicrobial properties against Staphylococcus aureus, against E. coli, and against Pseudomonas. But also we were able to, on this slide, but also we were able to kind of have this exopolymer, and we use them as a protective plastic or polymer for catheters. And we would have a catheter that we would infect with bacteria. This is the control. This is the infected catheter. And then when we did the infected catheter with the polymer protecting the catheter, we would see that we will not have any growth of bacteria. And also, we did other metals using this protective polymer. We grew zinc nanoparticles with this exopolymer. We grew nickel nanoparticles with the exopolymer. And then against resistant Staphylococcus and resistant Pseudomonas, we would see these antimicrobial effects, but also antibiofilm effects, which is really nice for our papers. For our therapeutic applications. And then finally, we did some of the really cool materials, which is combination of bimetallic nanoparticles. So we had these exopolymers, but then we put silver and iron nanoparticles so that we could develop materials that are antimicrobial, but are also magnetic. So, these are not nanoparticle. They are nanoparticles, but they are all agglomerated in a sheet of paper. But you can see that they are, they have these. This is the magnet, and so you can see that the nanoparticles have silver and also have iron, and they have metallic properties. And so then we did this iron nanoparticles, and we tested it against many different bacteria, and we would see that they will all have bactericidal and antimicrobial effects against different kinds of strains, Staphylococcus, Pseudomonas, E. coli, and even Candida albicans. And then we started working with a group in York University in England, and in the UK. And they are a group that produces cellulose, nanocellulose from from different waste products, especially orange peel. And so we wanted to see if we could have these biomaterials, make them therapeutical, and have the cellulose be kind of like a matrix for silver nanoparticles. So we actually did also the synthesis, and you can see here At least in this picture, you can see the nanoparticles, which are all part of this nanofibers of cellulose. And then we tested these nanocellulose nanofibers with nanoparticles, and we would see that in the control, you have the growth of the Pseudomonas and the Staphylos, but then at different concentrations, you would not see the growth of resistant Pseudomonas aeruginosa and resistant Staphylococcus aureus. So we had this really nice cellulose material, which is actually also antimicrobial. And lastly, I will talk to you a little bit about the new strategies that we are employing to find new antibiotics. And this is an idea that I got from my postdoc. I used to work with a really amazing now a professor at Brown University, Peter Belenky, and we had the idea that, you know, all our ecosystems are constantly fighting with each other, right? There's always someone that it's, even us as humans, right? We have our defense mechanisms to get food and to get the space, right? And some of these aspects are actually used in the food industry. For example, if you take a look at the brie cheese or salamis, all of this white stuff is penicillium, right? And they protect it with a lot of penicillium so that nothing else will grow in there, right? So it's impossible for a microorganism, or at least very difficult for a microorganisms to grow in the surface of these foods. And so this is actually a preservative or a way to preserve food. And this came from the very famous story of Alexander Fleming, which he found this, I guess, this microbial fight or this microbial war in a petri dish, right, where this penicillium 1 producing penicillin and killed these the Staphylococcus aureus. But I mean, this is not the only fight in nature, right? There's nature is full of these interactions, and so we started working in our lab. We found different strains of and types of microorganisms that we had in the lab, and so we started fighting Candida albicans with E. coli, and we had this really cool. I mean, this was our first fight. And our culture, and we had a positive result, right? So this is in these curves, you can see that we have E. coli growing, and then we have Candida albicans and E. coli growing. The Candida albicans grows nicely, but then when it grows in the presence of E. coli, you can see that around 4 hours you start seeing a decline in Candida albicans, right? So we hypothesized that E. coli was producing something. We did a lot of different experiments, and we came up with. We are currently working on the identification, but we came up with this culture that serves as an antifungal for Candida albicans, and it was actually the cover of Microbial Cells, which is a really nice journal. These are, you can see these are the Candida albicans, and these are the E. coli that are fighting each other and are producing this novel antifungal produced by E. coli. So then in my lab, what we did is we started also performing many of these kind of battles, and one of our protagonic roles was that Rhodotorula mucilaginosa strain that I talked to you about that produces EPS or exopolymers. And it was really cool because I thought that this exopolymer would be kind of like ammo or a warfare. It would allow these bacteria to protect itself from many things, metals, but also maybe bacteria, right? And you can see that when we grow the Rhodotorula in the presence of E. coli, you can see that it always as long as we give them enough glucose, it always produces an extra amount of exopolysaccharide. So this exopolysaccharide is actually has antimicrobial properties by itself, not just when we put silver and silver nanoparticles. So yeah, so you can see that it has antimicrobial properties against Pseudomonas, against Staphylococcus aureus is it's very notorious, its effect, some effects against E. coli, but especially it has antibiofilm properties. And this is very interesting because actually exopolysaccharides are produced to form biofilms. So it not only is used for Rhodotorula to form its own biofilms, but also protects Staphylococcus or Pseudomonas or even E. coli for them to produce their own biofilms. And we did a very nice toxicological effect on the exopolymer, and you can see that it's not toxic at all to human cells and even mice. So one of our ways of exploring is trying to join together engineering, the biotechnologists, the pharmaceuticals, and also machine learning and high throughput screening for us to be able to come up with like this predictive model where we can see that what kind of competitions will be more fruitful in the future to fight different infectious diseases and have kind of like our library of autochthonous strains or strains from other labs, etc. So with that, I finished my talk. I think that nanotech and biotech have a lot of different aspects that we can join from both of these fields, and we can have a huge advance in the development of new therapeutics. We know that nanotechnology, we're able to create very nice nanomaterials, at least at the nanoscale. But for us, it's very hard to go from nano to macro with this type of precision. And biology is just what it does, right? So you can see we are formed by the union of two half cells, right? Two half cells make us up, and that's the beginning of any human being, right? And just by physical and chemical interactions and its own biological program inside those cells, we create highly complex machines, which is us, right? And so I think that we can take a lot of learning from biotech and joining it with nanotech, so that we can create our own nanomachines, nanotherapeutics, with a lot of precision and different applications. So this is my research group. This is our research center. This is a picture from our lab where we had the visit of a Nobel Prize. These are some of my funding sources. I thank the university, CONACYT, which is the National Council of Science, which is the main funder for my group, but also the Secretary of Education and some of the Secretary of Defense. And these are my contact information if you want to contact me as well. Thank you very much.
Thank you, Professor Mezes, for this cutting-edge talk on antibiotics and nanotechnology. It's amazing, and it's an eye-opener. Would like to ask you a few questions about what's the affordability of these things at the moment? As you know, Yemen is a very deprived country and struggling with lots of medicines, let alone expensive antibiotics.
No, it's not expensive at all. Actually, if you're talking about these precious metals, silver, the amount of concentration that you need for an antimicrobial effect is very, very low. And actually, one of the main uses of silver, I don't know if you know, but silver was mostly used to reveal photographs. You know, when you go into that dark room, that liquid that the people put in their negative photographs is just silver nitrate. It's full of silver nitrate. From there, you can get, I don't know, thousands of just from that liquid, you can get thousands of antimicrobials developed in there, right? So now that people do not reveal their photograph so often, the use of silver has declined a lot. It's now mostly used as a precious metal, but also there's a lot of use as antimicrobial. So the price would not be expensive. Yeah, it's very cheap. And these, if you go from the aspect of the exopolymers or our biopolymers, they're mostly made from waste or from culture media. They're produced by bacteria. So also not expensive.
Okay, with regards to antibiotic resistance, what's your advice for our colleagues in Yemen? As you know, they often make diagnoses without proper cultures and sensitivities. They just go in empirical treatments. And I guess that's one of the main reasons why antibiotic resistance is driven up. And that's why we need to resort to cutting-edge technology such as nanotechnology to overcome the next wave of antibiotic microorganism resistance. So what's your advice for how to minimize the impact of inappropriate use of antibiotics in a deprived country?
Yes, this is also the case for Mexico, right? The clinic is very, how you call it, is very symptomatic, right? So a doctor, doctors in Mexico, very few times, at least in your first visit, they don't do any tests, right? They say, oh, you have a fever, they see your throat, and you have an infection, right? Or it's very very European in the French school, right? Where there's through symptoms you prescribe something. Very different from maybe the American way, right? Where the Americans, you go with whatever, as small as you think it is, maybe a small fever, etc. They do many tests, right, to be sure what they're going to do to you. And the United States and many of other Occidental countries, in regarding infections, they do these antibiograms, like you mentioned here in the Q&A, and they don't give you any antibiotic until they're sure which bacteria you have and what is that bacteria sensitive to, right? Which is the very nice way of doing it. But sometimes you have such bad infection and such horrible symptoms that waiting a couple of days for these antibiograms to give you a result, sometimes it's very difficult to do, right? And also it's very expensive to keep doing these things over and over again. So I guess my advice is to do more antibiograms, right? I mean, to only give this kind of antibiotics when you are almost sure it's an infection, and you can say, in this case, I'm not going to do an antibiogram because I am sure I can see the infection. But most of the time, people doctors in Mexico give antibiotics, even though here in Mexico there is a law about 6 or 7 years ago that came out that you cannot have antibiotics over the counter, because before you could buy antibiotics over the counter. So now they have to be administered by a doctor. But still, doctors have this I don't know how to call it, but they have this custom to at least give you antibiotics at the smallest of symptoms. So that's really, really bad. On the side of all of the antibiotics that are used in agro, food processing, etc., right? It's not just the clinic. I don't know if I answered the question, but I don't have a very good advice for that.
Thank you. It's a challenge because of the lack of resources on the ground, lack of testing, and sometimes also pressure from patients as well.
It's also cultural, right? So here in Mexico, if a mother goes with his child and he has a fever and the doctor doesn't give him an antibiotic, they're like, he needs an antibiotic, right? Even though, so she fights until the doctor gives them an antibiotic, right? Because she doesn't feel that it's that it was the right treatment, right? So, or go to another doctor who gives them the antibiotic. So,
Do you think the battle against the bacteria will be won with nanotechnology?
the battle against antibiotics, in my perspective, will never be over, but it can be controlled, right? I mean, you can think about bacteria reproduce every 30 minutes. Every 30 minutes, they have the ability to produce one member of its huge population to be resistant to any weapon that you give it, right? So it's very hard for us to say that we are going to eradicate bacterial infections at any point, at least from my perspective now. So it's just a way of us to say we will never win this battle, but we have to constantly come up with new ways of combating bacteria. Right now in my group, I didn't want to show this because it's not over yet, and we haven't published, but we are working with antibiotics that are a little bit more intelligent and are, we call them cyclic, right? So they're intelligent because of the delivery. We're working with live bacteria inside biological materials. And so these bacteria are triggered by a pathogenic bacteria, and then they produce an antimicrobial or an antibiotic. They produce it, and then when the bacteria goes to a certain threshold, it stops producing it, and then it goes back and it starts until it keeps it at a threshold where the body can eliminate the bacteria. So in that way, bacteria will never really become resistant because you don't really have that pressure, right? It goes back. It's kind of like a cyclic antibiotic. So we're trying to see if we could combat resistance through these methods, right? So hopefully we will be successful.
Thank you very much. There's a question by Shihab Sahabani. Unfortunately, we don't understand what exactly your question is. So if you can rephrase your question, we will happily pass it on to our guest speaker. While we give Shihab a chance to rephrase his question, we'd like to ask you about in terms of other areas to combat against antimicrobial resistance. So we have the nanotechnology on one side. Is there any other frontiers in science that also are trying to combat that?
Yeah, I mean, there are many ways to, not just nanotech. I think synthetic biology, biotechnology, understanding how bacteria evolve, that's going to the principles, right? You can find a lot of information right now, even though we know how antibiotics work. For example, penicillin, right? There are many things that we still don't know about it, right? What are the mechanism of actions? Once we know about mechanism of actions and the biology of it, we can do all these things that I try to do, which is trying to come up with combinations of antibiotics that have synergy, that prevent resistance, right? So I think it's not just, nanotech gives you a lot of tools to build these kind of like nano machines or materials that are intelligent, right? But or that are dynamic more than intelligent. But the resistance of bacteria, I think, has to go back to the principle of biological evolution, of antimicrobial mechanisms of action of what we have right now, for us to be able to kind of utilize our resources much better, right? So
thank you very much. Thank you very much. I don't think there is any more. Let me just have another check there. All right, okay. So this is a question about water filtration using silver kind of immigrated products for kind of trying to make antiseptic. Question is, silver is used for filtration? I guess it is. I think it is, but I'm not sure of.
Yes, I mean, there are different regulations in different countries. For example, here in Mexico, we have a product called Microdin, and Microdin is basically colloidal silver. It's not nanoparticle silver, it's just colloidal. It's made by some different synthesis. So the silver particles are much larger, and they are used to treat many foods, especially vegetables that are grown in the ground. For example, strawberries, lettuce, broccoli, right? So what we do is we put those fresh produce in a bowl of water, and we add a couple of drops for 10 minutes, and that eliminates any pathogen that might be in the produce, right? But I don't know if in other countries they have these kinds of products. Silver is also used a lot in especially the supplements market. So the supplements market, as you might know, it kind of like takes a shortcut on all of the FDA approval, etc., and you take it on your own terms, right? So there's a lot of silver products involved in supplements, which people take, and there has not been, I mean, there isn't a very thorough experiments done on this, but it's in the market, and it hasn't caused this huge amounts of deaths, etc., right? So I think in perspective, it's not such a toxic chemical in its right proportions, right? And also silver is used in many plastic containers, in textiles. A lot of silver threads are added to textiles to prevent bacteria growing from which cause bad odor. So a lot of the textiles for sports or for athletes, they are some that have silver threads that prevent these bad odors from bacteria growing in the textiles from sweat.
Great. Thank you very much for that. I hope we addressed this question to our colleague Shihab Sabai, who's asking this. One final question. In a low resource country like Yemen, where you don't know what is the exact pathogen, and you risk already an existing resistant pathogens, do you recommend monotherapy or polytherapy, like multiple antibiotics to treat the pneumonia, for example?
It's very tough. It's a very tough question, and it's actually a very big problem, because I don't think it's just Yemen. I mean, obviously, countries that have lower resources in their for their medical supplies, but it's hard to say. What I'm mostly scared is the fact that there are some studies that say that when you treat bacteria which are resistant with an antibiotic, the culture from that bacteria, since at least you don't kill the bacteria, but you mess with it, right? You can create an environment for multiple mutations, right? So when you take an antibiotic and you do not kill the bacteria, you might form cultures that are resistant to other antibiotics that were not previously that this culture was not previously resistant to, right? So it is very hard. It is very hard to. I think my recommendation is that at least just, and I'm not a doctor, right? But just from common sense, if you have an infection on a site that is very hard to reach. For example, if you have an internal infection, if you have an infection on a catheter, if you are in the hospital, it is always better to do an antibiogram, right? Because in hospitals, most of the bacteria are multi-resistant, because they are in the hospital and they have been treated many times by many different antibiotics, and the people are there being treated. So I think that's the best bet. Going on a coin flip and saying, I'm going to give you this antibiotic, even though I don't even know what bacteria you have, it's basically a coin flip. Yeah. So it's just luck.
Yeah, I think, as you said, in a place like this, common sense is very important. Like you said, if there is, if it's a hospital-acquired infection that is deep infection, most likely to be resistant, maybe multiple antibiotics, especially if it's, or even if it's a patient who can't take a risk, for example, patient in ITU and intensive care, a patient who is immunosuppressed or compromised.
Yes.
I really thank you very much for this talk and coming to us all the way from Mexico and joining us from there and giving us your insight on this cutting edge technology and how a place in Yemen can look forward to maybe improve their ways of antibiotic stewardship in the future as well. I don't think we have any more relevant questions apart from some of them which we have already touched on. There are also no other questions for the other speakers because Dr. INTERYem has kindly answered the questions left over by typing and directly messaging people who asked on the Q&A session. I'd like to thank all our guest speakers for being with us today. I'd like to thank them for the time and for the immense knowledge they shared with us today. It's an eye-opener going from COVID, transmission, biology, treatments, vaccinations, everything we needed to know about vaccinations today was touched upon. And by Professor Tang, we also had a really thorough explanation of the situation on the ground with regards to COVID surveillance and really the challenges with regards to resources and compliance and also accurate reporting, which will be an ongoing issue. And then we finished off by a talk by Professor Ramirez with regards to antibiotics challenges and opportunities, touching on a lot of the coming technology, which will be our weapon with regards to the next wave of antibiotic resistance. What we'd like to say is all these are for people of Yemen, our colleagues and friends there, to be connected to the world. So if you have found anything that is useful here, you'd like to get in touch with relevant people abroad to mentor you and support you, we'll be happy to take information via INTERYem website, contact us via the email there with your proposal and your ambition, and we can link you with relevant people because this is the most important thing, is creating a window for people in Yemen to network with the outside world. Look forward to tomorrow, which will be full of equally as important talks from everyone. In the meantime, enjoy your evening and look forward to have you again tomorrow with us. Assalamu alaikum wa rahmatullah.